several proteins for the subunits of the respiratory chain and nuclear DNA genes. Mutations in those genes are responsible for the mitochondrial diseases. These are a group of syndromes and diseases that are very heterogeneous, and they are caused by both nuclear and mitochondrial DNA mutations. They affect children and adults. There is no epidemiological studies in Brazil, so the estimated prevalence of these diseases is about 12 to 100,000 adults. This is from a study performed, done in the northeast of England. The clinical manifestations arise from different organs, as demonstrated here. But the most affected organs are the brain, skeletal muscle, heart, and the eye. The mitochondrial DNA is a circular molecule with about 16.5 kb and with 37 genes encoding 13 polypeptides that are subunits of the respiratory chain complexes, 22 tRNAs, 2 ribosomal RNAs. It's important to remember that only the minority of the subunits of the respiratory chain complexes are actually encoded by the mitochondrial DNA. The rest is all from the nuclear DNA. Here we demonstrate in blue, red, green, and yellow, the ones that are encoded by the mitochondrial DNA. And it's relevant to know that only complex 2 has all the subunits encoded by nuclear DNA. The defects of mitochondrial DNA can be classified in 2 types: large-scale deletions, large-scale rearrangements, which are sporadic in nature. The most frequent is large deletions, and the more rare is the duplication. And point mutations, which are usually with maternal inheritance. This is an example of a deletion. You have the normal mitochondrial DNA here in a blot, and the band with loss of a large fragment of the mitochondrial DNA. And this is the duplicated mitochondrial DNA molecule. When the point mutation affects ribosomal RNAs or tRNAs, the protein synthesis is compromised, and we have a combined deficiency of all of the complex respiratory chain complexes with subunits encoded by the mitochondrial DNA. When there is a point mutation that is specific to a subunit, only one of the complexes is compromised. Large deletions, they affect a large segment of mitochondrial DNA and involve several genes. So the defect is defecting all of the complexes encoded by mitochondrial DNA. There are also secondary mutations in the mtDNA that are due to nuclear gene mutations. Those are multiple deletions, which are both multiple deletions and depletion with Mendelian inheritance. Multiple deletions are several deletions with different sizes that we can see here. This is the normal and several smaller bands. Depletion is a quantitative defect when we have a decreased amount of mitochondrial DNA molecules in the mitochondria. So the small amount is not sufficient to provide the energy to the function of the mitochondria. And this is the representation of the blood with a patient with a depletion of mitochondrial DNA. When we talk about mitochondrial DNA, we have to have in mind that there is the situation of heteroplasmy when we have a mixture of mitochondrial DNA with mutation and normal mitochondrial DNA. The proportion of mutant mitochondrial DNA is important for the phenotype, for the manifestation of disease. Higher proportions of mutant mitochondrial DNA will have higher chances of developing the disease. This is a schematic figure, a cartoon, demonstrating how we see in a patient the heteroplasmy. A patient that is most affected has higher proportions of mutant mitochondrial DNA in different tissues, and so he has manifestation like diabetes, heart block, weakness, and so on. And the one that has lower proportions of mutant mitochondrial DNA does not develop the disease. Mutations were described all over the entire region of mitochondrial DNA sequence with different phenotypes. Here you can see examples of phenotypes described in mitochondrial DNA mutations. You can see that we have, here's an encephalopathy, cardiac involvement, myopathy. This is ophthalmoplegia, ocular myopathy, diabetes, deafness, gastrointestinal dysmotility, anemia, dementia, and so on. Interestingly, in one phenotype, for instance, in myopathy, we can find different mutations causing the same phenotype. And at the same time, one specific mutation, for instance, this one, the 3243 mutation, appears in several different phenotypes. So this is what makes so difficult sometimes to make a clinical diagnosis. One thing is very interesting in mitochondrial disorders is that in muscle we can find this type of fiber with increased amount of mitochondria. We call mitochondrial proliferation. That in the staining it's colored in red. It's usually called as red fibers. This fiber can be seen in histochemical reactions. This is STH, succinate dehydrogenase, which is complex 2, cytochrome C oxidase, complex 4, and here's a double staining. Here we can see the increase in intensity of the staining due to the increased amount of mitochondrial proliferation. Because complex 2 is not affected by mtDNA mutation, this is preserved, the activity is preserved. On the other hand, complex 4 has a deficiency, is deficient in the same fiber. These are zero sections, so these are the same fibers. This is very typical of a mitochondrial DNA mutation. Using staining, we can also see that a subunit 2, the subunit 2 of COX, cytochrome C oxidase COX-2 subunit, is deficient, is less expressed, has a decreased expression in the muscle with mitochondrial proliferation. But the subunit encoded by the nuclear DNA, that's COX-4, is preserved. These abnormal fibers have usually increased amount of proportion of mutant mitochondrial DNA. We can see this easily isolating single fibers and analyzing the proportion of mutant according to the COX activity. So we have isolated fibers with mitochondrial proliferation and COX deficiency and COX normal fibers. And we can observe the higher amount of proportion of mutant mitochondrial DNA. Due to the easy access of NGS, next generation sequencing, more nuclear genes have been identified in the last years. We can group these nuclear genes affected in mitochondrial DNA as those affecting the structure of the respiratory chain. Those are subunits, genes encoding for subunits of the complex. Factors affecting the assembly of the complex. Factors affecting mitochondrial DNA synthesis. Those affecting protein synthesis, and others like coenzyme Q biosynthesis. The complex the respiratory chain complex has several subunits. Most of them are synthesized in the cytoplasm and has to be transported to the mitochondria, and then they have to be joined with the other subunits to form the entire complex. Any mutation in any gene encoding for the factors participating in this process can lead to defects in assembly complex. And then complex 1 deficiency, for instance. At the same time, the complex can be joined together. For instance, complex 4, 3, and 4 are forming a large complex called respirosome. And anything happening during this process is also leads to respiratory chain deficiency. This is an example of a patient with a nuclear gene defect. This is a muscle biopsy where we can observe that there is a deficiency in cytochrome C oxidase activity compared to the control. The same deficiency can be observed in fibroblasts, and using fibroblasts we can analyze other features. Here we found that complex 1, complex 4, and complex 5 were not assembled. There was a decrease mitochondrial protein synthesis, but the transcription was normal. This patient had mutation in this gene, RMND1, which is important for the protein synthesis. Another pathway that is affected by several mutations is mitochondrial DNA synthesis. This is a scheme showing the several steps involved in the process. And the colored balloons are the genes that have mutations already associated to patients. I can highlight TK2, which is thymidine kinase 2, responsible for the conversion to the deoxyribonucleosides, polymerase gamma, which participating in the replication process, and all of them give mitochondrial DNA deletions or depletion of mitochondrial DNA. Until now, there is no curative treatment for these diseases. The treatment is only symptomatic. We usually treat like epilepsy with the anticonvulsants, diabetes. When there is a coenzyme Q deficiency due to a problem of the biosynthetic pathway, the supplementation is recommended. But other than that, there is nothing more effective. But there are several ongoing studies with several strategies. For instance, some try to increase mitochondrial biogenesis, increasing mitochondrial content. They try to compensate for the energy deficiency. Gene therapy for MD4 mutations, especially for those affecting the optical nerve. Nucleoside bypass therapy for the TK2 mutation. Several antioxidants are being tested, considering that the respiratory chain when defected will increase the production of reactive species, and mitochondrial DNA editing strategy where it's tried to cut the mitochondrial with the mutation and keep only the wild type mitochondrial DNA. In 2015, the Mitochondrial Medicine Society recommended this treatment, the supplementation with arginine to this syndrome called MELAS. MELAS is a syndrome that stands for mitochondrial encephalopathy, lactic acidosis with stroke-like syndrome. The patient usually have episodes similar to a stroke, but it's not a real stroke because there is no obstruction in the vessel. The patient also have epileptic seizure or dementia, lactic acidosis, the ragged red fibers, and it's commonly caused by the 3243 mutation. In the muscle vessel, intra-arterial vessel, it was observed that in the wall of the vessel, the same abnormality with increased mitochondrial content was seen by the succinate, the SDH histochemistry. Here is a vessel from a patient, and this is from a normal patient. And by electron microscopy, we can see that there is higher number of mitochondria in the wall of the vessel. So there are two main hypotheses to explain the stroke-like episodes in MELAS. The first is that it's an angiopathy causing an inappropriate intracranial hemodynamics during the stroke-like episodes. And the second is that it's a metabolic cytopathy causing an imbalance between energy supply and demand and provoking neuronal hyperexcitability or damage. This therapy was proposed by a Japanese group in 2002, Koga et al., thinking that the stroke-like episodes were an impairment of the vasodilation during that episode. They thought they administer L-arginine, thinking that L-arginine would induce the production of nitric oxide, and nitric oxide would induce vasodilation, and so improve cerebral blood flow. And in fact, in their studies, they see several improvement in some clinical manifestations, such as headache, nausea and vomiting, and some visual manifestations. But more recently, a systematic review did not find any clinical benefit for this treatment, even though everybody is using now for the patients. So we reviewed several articles trying to see what's known about the mechanisms regarding L-arginine in millers, and we found that the studies had very small number of patients, but it's clear that there is a deficiency in L-arginine in patients. There is a low level of arginine in the patients with MELAS. But the mechanism seems very complex, not only affecting the blood flow, but arginine has been reported as acting in stabilizing complex 1,3 supercomplex, acting in increased TCA cycle activity, reducing lactate, reducing nitrooxidative stress, and increasing neuronal activation. So more data is still needed to clarify this part of the mechanism. There are several challenges in terms to find a treatment to mitochondrial diseases. I highlighted those three. First is the rarity of the disease, which makes it difficult to form adequate number of group of patients. The patients are heterogeneous, so it's really difficult to have homogeneous groups. And there is a lack of robust animal models that mimic the disease. And I brought these two graphs to show how heterogeneous these patients are. These are marker of nitrooxidative stress. The patients are discriminated by the colors. Blue are the normal ones, and each dot is one fiber in the muscle. So we can see that here in this mutation, which is mtDNA encoded mutation, mtDNA gene, we have a large variation among the patients, and even in one patient in the muscle, there is also a large variation. And when we see another gene, which is nuclear, it's more homogeneous, but still there is one patient that stands out. And it seems that there are other factors interfering with the expression of the disease here. And these factors are still not known. And I want to thank the financial support, and all this work was done with the group of neuromuscular disorders at Escola Paulista de Medicina, and with the collaboration with Carlos Moraes, the University of Miami, and Michio Hirano, the Columbia University.
Thank you so much for the wonderful talk. Any questions?
Thank you. Great talk. My question is related to the immune system of these patients. Usually, some of these patients, they have infections quite often. So do you know how is the heterogeneity threshold in immune cells of these individuals? Because it's quite common. Usually we focus on like solid tissues, but not like the immune cells.
I don't know. I didn't see anything focusing on the immune cells. What we really know is that no, it's they get worse when they get infections. That's all. I don't know if they have deficiency in the immune cells. In the hematopoietic system, we only know that there is one form, that is Pearson syndrome, that compromises the red blood cells and causing anemia. But we don't have any other manifestation.
Thank you.
Congratulations, Professor. My question is about the perspective of the future. How will you see the perspective of the using of the CRISPR-Cas9 on the treatment of these diseases?
I don't have any experience on that, but what I heard is that it's more difficult to get into the mitochondria. That's why they try to use mitotalin. It's another technique, but it's easier to go inside and get the target correctly.
Thank you. I do have one question about the arginine. You know, arginine is a potent activator of mTORC1. And if you have any evidence that could be somehow the mechanism by which arginine is doing some, improving some of the phenotypes that you're seeing.
Yeah, as I said, I didn't find anything about mTORC, but as I said, we don't have any evidence of the effect. We don't know if it's really improving anything, because when the patients get the stroke-like episode, they also have seizures, and they also receive anticonvulsants, so epileptic drugs. So we don't know. Some authors think that the effects are only for the anti-seizure drugs, not due to the arginine. And it's difficult to see the effects, because the studies are small, and it's even harder because you have to treat on the acute attack.
Yeah, thank you for your talk. It was really interesting. If we are looking for patients with mitochondria disease and with mutation in the mitochondria genome, usually they get multiple mutation. So don't you think that's a problem to find a treatment, and that probably we will need to go to more to genome editing for the mitochondria genome, and probably for the patient to receive corrected mitochondria, hoping that this corrected mitochondria will correct the phenotype?
Yes, genome editing would be perfect, but you have to think that the disease is rare. For instance, the most frequent point mutation is that 3243. We don't have any editing strategy for this mutation. No, I need to correct. We have one lab working on that, but it's difficult because we don't have the animal model for 3243 mutation, the reliable animal model, and it's rare. This is the most frequent. If you will have to work in each mutation, so it's very expensive.
Thank you.
Thank you so much. I think the next speaker is me.
It's my pleasure to introduce Professor Sovan Sarkar. He's an associate professor at the University of Birmingham, UK, and holds the distinction of former fellow for life at Hughes Hall, University of Cambridge. He did his PhD from University of Cambridge and postdoctoral research at the Whitehead Institute in the USA. Utilizing human pluripotent stem cell-based models, his research focuses on the role of autophagy in cellular homeostasis and survival, and biomedical implications of this process in neurodegenerative and lysosomal storage diseases. He's also the head of global engagement for the School of Medical Sciences at the University of Birmingham and holds a collaborative University of Birmingham Brazil Institute USP seed fund with Edesco Neto, with me. Thank you very much, Safa. What's yours?
Thank you, Odysseus, for organizing this conference with such wonderful speakers and amazing venue. I like Brazil. This is my 7th trip, and I like to collaborate with people here, and I also host a number of Brazilian researchers, including undergrads to visiting professors as well. And now in my new role as a global engagement head, I'm happy to assist you if you are interested in collaborating with Birmingham. So I primarily work on autophagy, but the good news is I have some mitochondrial data that I will show you. So it won't be that boring maybe. So first, although I'm sure you know about autophagy, I'll just give some basic overview that it is an essential cellular process which is critical for the homeostasis and survival of the cells, and therefore it is important for human health. So this process, happens in the cytoplasm of a cell, where a membrane structure called phagophore is formed primarily from the ER, and as it engulfs, it captures cellular rubbish. These are undesirable materials such as damaged organelles such as mitochondria, aggregated proteins, and so on, and they get engulfed into this double membrane vesicle called autophagosomes, which then fuse with lysosome to form autolysosome, where the cargo is degraded, and this helps in maintaining cellular homeostasis by removing this cellular rubbish, and therefore the primary role of autophagy is housekeeping, clearance of garbage. Okay, and this is mostly it can be selective or non-selective. Now, an additional feature is metabolic homeostasis. So many of this cargo are macromolecules such as proteins and lipids. So therefore amino acids and fatty acids that are generated after degradation of this cargo enters into different biosynthetic pathways to generate energy. For example, starvation is a classic physiological inducer of autophagy. So if you're starving, you're inducing autophagy. Therefore, the cells are eating their own macromolecules to generate energy so that your cells can survive. Now, because of this critical role of autophagy is implicated in a variety of pathological conditions ranging from aging and cancer to myopathies, liver disease, as well as neurodegeneration. But beyond disease scenarios, autophagy is critical for development, immunity, longevity, and so on. And my talk is focusing on the role of autophagy in neurodegeneration. Now, first, I'd like to highlight two classic papers that were published in Nature in 2006 from Japan. So what they did is they took normal mouse, they selectively deleted essential autophagy gene in the brain of normal mouse, which developed neurodegenerative phenotype. So this suggests that basal autophagy is critical for neuronal homeostasis and health of the brain, whereas loss of autophagy causes neurodegeneration, implying that autophagy dysfunction in neurodegenerative diseases could be a causative factor for neuronal cell death. But beyond brain, there has been multiple studies between 2006 and 2010 where there has been tissue-specific deletion of autophagy genes, such as if you delete autophagy in the heart, muscle, or liver, you see tissue-specific degeneration in the mice. So therefore, overall, autophagy is important for tissue homeostasis and so on. So there are many autophagy substrates such as long-lived proteins, as I mentioned, damaged organelles such as mitochondria, aggregated proteins, even lipids and cholesterol, also pathogens such as bacteria and viruses. And focusing more on the aggregated proteins, a number of these proteins associated with neurodegenerative conditions such as mutant Huntington's in Huntington's disease, alpha-synuclein in Parkinson's are predominantly degraded by autophagy because these large structures cannot be degraded through the narrow proteasome barrel. So we and others in the past have shown that chemical or genetic induction of autophagy can enhance the clearance of these mutant proteins, leading to reduced protein aggregates and protection in a variety of transgenic models ranging from flies to mice. Now, the critical pathway that regulates autophagy is mTOR complex 1. We heard about it in previous talk. Now, activation of mTOR inhibits autophagy, and there are multiple upstream signals all impinge on mTOR to negatively regulate autophagy. Now, this classic paper was published back in 2004, and this is a lab in Cambridge where I did my PhD, and they are the first one to show that if you inhibit mTOR with rapamycin or its analog CCI779, you can induce autophagy which can enhance the clearance of mutant Huntington. And this was shown to be protective in Drosophila and mouse model of Huntington's disease. So this is the first paper showing a biomedical benefit, therapeutic benefit of autophagy induction in an animal model. Now, while this is very exciting, I'd just like to highlight that mTOR has critical cellular pathways such as cell growth and translation. And therefore, you might not want to use mTOR inhibitors. They are also immunosuppressants for long-term drug administration in neurodegenerative patients. So previously, during my PhD, we identified the first mTOR independent autophagy pathway, meaning that you can now regulate autophagy without affecting mTOR. So the first pathway we identified was by inositol and IP3 levels, where increased in intracellular, these agents can inhibit autophagy. And in this pathway, we found that mood stabilizing drugs such as lithium, carbamazepine, and sodium valproate can all stimulate autophagy by lowering inositol and IP3 levels, and to stimulate mTOR independent autophagy, which we and others have shown to be protective in a number of animal models of neurodegeneration. Now, beyond this, we did a number of additional studies and chemical screen to identify a large network of mTOR independent autophagy pathway, where I'm just highlighting some of the key inhibitory signals, such as increase in cyclic AMP. I already mentioned about IP3, also increased calpain activity, increased nitric oxide, and so on. And in these pathways, there are multiple autophagy inducing compounds that we identified, and the list has grown since then. These are very old, like more than 10 years ago, but some of these drugs are FDA approved, such as verapamil and so on. So now we have a large database of autophagy inducers that one can use for basic research or biomedical research for inducing autophagy without the need of using rapamycin. Now, as I mentioned, that knockout of autophagy in mouse brain can cause neurodegeneration, and therefore, indeed, autophagy impairment has been demonstrated in multiple neurodegenerative disease, ranging from Alzheimer's to several polyglutament diseases such as Huntington's, spinocerebellar ataxias, and so on. But this is just a small list. The current consensus is if you study autophagy in any neurodegenerative condition, you are likely to find a dysfunction. That's how it works. Because autophagy declines with aging and neurodegeneration. Now, apart from these common, diseases, I would, want to discuss about defective autophagy in certain rare early onset, like childhood onset neurodegenerative conditions, as well as in certain rare lysosomal storage disorders. And why this is our interest is because, I will just mention, sorry, I just skipped some slides, because we, as part of Center for Rare Disease at the University of Birmingham, where I'm collaborating with Tim Barrett, who is the director of the center, one of the objectives is to target defective autophagy as a shared pathomechanism. So we want to target, we want to establish this autophagy defect in multiple rare disease, and therefore come up with a unified therapeutic solution so that in future we can do a clustered trial with repurposed autophagy-inducing drugs. So in my lab, we are primarily, focusing on human pluripotent stem cells to, undertake human-relevant biology, and we also collaborate with, other people for in vivo work. So typically, as you all know about iPS cells, you can now generate iPS cells from patient fibroblasts, skin fibroblasts, or blood cells, and then differentiate them into the desired cell type. You can also correct, the mutation to create an isogenic control by genome editing, and we are mainly focused on disease modeling as well as drug screening to identify therapeutic compounds that can be hopefully taken to clinical trials in future. So the two diseases I'll be briefly highlighting are one is a Niemann-Pick type C1 disease. It's a cholesterol storage disorder. And previously we showed that it is impairing autophagy at this autophagosome maturation stage. It prevents the fusion between autophagosomes and lysosomes. And another disease called Wolfram syndrome. It's also a childhood onset neurodegenerative disorder, also associated with diabetes, where the defect is at a different stage. It prevents the formation of phagophores. So in either case, it's preventing autophagic flux and impairing autophagy. So we have previously published, the autophagy defect in Niemann-Pick disease, where we found that this mutant protein, loss of this NPC1 protein, which is located on the late endosomal and lysosomal compartment, actually prevents the vesicle fusion by failure of the snare protein complex. So the snare complex between VAMP8 and syntaxin 70 is important for this membrane fusion, where loss of this NPC1 protein prevents this interaction. Interestingly, we found that pharmacological induction of autophagy can synthesize new autophagosomes that can rapidly fuse with lysosomes to clear the cellular rubbish and restore autophagic flux. So that was interesting in the context of a lysosomal storage disorder. So again, some old data where I just want to highlight some of the data in terms of disease modeling in iPS cells, we generated hepatic cells and neuronal cells because liver and brain are the two affected organs in the disease, and all the cell type characterization have been published previously. And interestingly, we see increased cell death in patient hepatic cells and patient neuronal cells compared to control. Now, why this is important? Because we are seeing this cell death phenotype at a basal level without the need for adding any external stressors. Often, in late onset diseases such as Parkinson's and Alzheimer's, iPS-derived neurons do not show this phenotype, whereas we are seeing this phenotype in early onset rare disease. So hopefully, the idea is to make discoveries in this rare disease platform and take it to more common, late onset diseases. Now, apart from this cell death, we see impairment in autophagic flux. So again, here, LC32 is a marker for autophagosomes, and increased LC32 in patient hepatic and neuronal cells means there's an accumulation of autophagosomes. Now, this could mean an induction of autophagy or impaired maturation or impaired, autophagosome lysosome fusion. And there is no single assay for doing autophagy. You have to do 3-4 assays to understand what's happening with autophagic flux, but a simple way is to test P62, which is a specific autophagy substrate. So if autophagic flux is impaired, P62 accumulates. So if autophagy flux is increased, P62 gets decreased. So here, apart from this and many other data as published in the paper, we confirm that there is a serious block in autophagic flux. And we also did genetic correction to fix the mutation, which could also fix the cholesterol problem and the autophagy problems and the cell death problems, which I'm not mentioning here as it's published before. But just want to highlight that some of the autophagy inducers that we identified in the past, we applied in this patient neuronal cell and hepatic cell, and we found that all the autophagy inducers that restored autophagic flux could rescue cell death, but only two of them, rapamycin and carbamazepine, were selective in rescuing autophagic flux, whereas verapamil could not fix the autophagy problem in hepatic cells. So we are using here rapamycin as a control, like it's an mTOR inhibitor, but carbamazepine is a mTOR independent autophagy inducer that could be repurposed for future. So this is interesting. Now, of course, then in our hands, we find that autophagy induction is cytoprotective in Niemann-Pick patient neurons. Now, follow up a study with a collaboration at the Broad Institute in MIT, where they screened 60,000 compounds in HeLa cells to come up with 5 strong autophagy activators, which we then tested in Niemann-Pick patient-derived neuronal and hepatic cells. So what we found is interesting. So this is, I'm just showing the cell death phenotype. So this is, you see the second bar is cell death in patient neuronal cells compared to control. Again, you see more cell death in patient hepatic cells compared to control. Carbamazepine, the mTOR independent autophagy inducer, is used as a positive control from our previous study. Interestingly, we found that these 5 amazing autophagy activators in HeLa cells, only 3 of them working in neurons and none of them working in hepatic cells. So I'd like to highlight that there is human cell type specific efficacy or mode of action of autophagy inducers. And even if we saw that even if something is working in mice, they may or may not work properly in human cell type, disease affected cell type. So I think it's, although in vivo studies are extremely important, it's also, having this iPS-derived preclinical platform is also a good idea to do. So next, I want to just, briefly highlight the other, the rare disease I am talking about. And you can see that how we are developing a common treatment strategy for 2 distinct rare disease. So again, this is Wolfram syndrome. It's also associated with, apart from neurodegeneration, it also has epilepsy and diabetes. And here, as I mentioned, that instead of accumulation of autophagosomes, we see the opposite. We see decrease in autophagosomes. As you can see by decrease in LC32, you can see the puncta, the autophagosomes are reduced. Now, again, we are confirming the problem in autophagic flux by accumulation of P62. So accumulation of P62 is a constant feature of defective autophagic flux, no matter which mechanism autophagy is being impaired. Now, the next interesting part is you'd expect, because autophagosomes are decreased, you'd expect these autophagosome precursors, phagophores, would be decreased too. But instead, we see there is accumulation of phagophores, as you can see with these phagophore markers. And so to understand the mechanism, we did a quantitative proteomics, and we found that regulation of macroautophagy is one of the significantly affected Go biological pathway. And we identified ATG9A. It's one of it's an essential autophagy protein that helps in the transfer of lipids. So you see this is a double membrane structure. ATG9A helps to deliver lipids so this structure can grow to form autophagosomes, and because this is depleted in Wolfram cells, therefore this is associated with accumulation of phagophores. And we further confirmed our proteomics data by Western, and you can see massive depletion of ATG9A by Western and Immuno. And interestingly, we saw that if we overexpress ATG9A in Wolfram cells, we can now rescue the autophagy phenotype. Like we can now increase autophagosomes, and now we can reduce this immature phagophores, meaning now the phagophores are being converted. And this is the mechanism. I'm not going too deep into it. We are seeing that the WFS1, the protein that is mutated in Wolfram syndrome, also called Wolframin, interacts with ATG9A to stabilize the protein that helps to deliver lipid for this phagophore elongation and closure. But mutation in WFS1 cannot stabilize ATG9, which probably gets rapidly degraded by this E3 ubiquitin ligase HRD1, which we are currently confirming. Now, moving on to disease modeling using the IPS platform. So we are collaborating with Laetitia Aubry in France, where we got 2 control lines and 3 patient lines, and these are some neuronal stem cells or neural precursors. These are some cortical neurons, and you can see there's expression of WFS1 in the control cells, but nothing in the patient cells because mutant WFS1 is very unstable. And in this system, we again confirm that there is reduction in autophagosomes as seen by reduction in LC32 in both neural precursors and neurons. We also confirmed reduction in ATG9A, and interestingly, just like in Niemann-Pick, we see that Wolfram patient-derived neurons have more cell death compared to controls. So next, we also found mitochondrial dysfunction here, and this is published. So I'm just giving a brief overview. Our transcriptomics data identified a number of genes associated with mitochondrial electron transport chain, and this was some of them were common between neural precursors and neurons, and we further went on to confirm a mitochondrial defect that these Wolfram patient-derived in neuronal cells and neural precursors are associated with mitochondrial depolarization, increased oxidative stress, and less ATP levels. So next, we did a genetic rescue experiment, and in this scenario, we are using a mutant cell line where from the AAVS1 locus we are overexpressing the wild-type protein under doxycycline inducible promoter. So what happens here is that if you add doxycycline, you can see restoration of the wild-type protein. You can now see WFS1 is expressing in the neural precursor and neurons, but if you don't add dox, nothing happens. They stay as mutant line. Okay, and so we see that addition of dox can rescue can restore WFS1 levels and rescue the autophagy defect, as you can see by increase in autophagosomes and ATG9, and also here you can see it can rescue cell death. So if I draw your attention to this graph, we are using a control cell line. If you add dox, nothing happens. This is an uncorrected mutant line, again, which has more cell death, but dox doesn't have any effect. And this is the genome edited rescued line where you add dox, you can see the cell death rescue. So meaning that this restoration of the wild type protein is actually rescuing these phenotypes instead of any non-specific effects of dox. So next, we wanted to test, pharmacological. We also, showed mitochondrial, correction. Like, this genetic rescue can also restore mitochondrial membrane potential. It can also rescue oxidative stress and, improve, cellular bioenergetics. So next we wanted to test, the pharmacological inducers of autophagy, and again as a proof of principle concept, we are using rapamycin as a mTOR inhibitor and carbamazepine as an mTOR independent autophagy inducer. And here we see that in patient neurons have less autophagisms and accumulation of autophagy substrate, which by addition of autophagy inducers get reversed, meaning that these inducers are now restoring autophagic flux, and this was associated with also clearance of agrosomes. So agrosomes are accumulation of misfolded protein, aggregated proteins, which can be visualized by this dye called proteostat. And you can see this proteostat intensity is very high in patient neurons, which is rescued by the autophagy inducers. And associated with this, we kind of see that the mitochondrial membrane potential, which is depolarized in the patient neurons, is also restored. So in the last section of my talk, I will kind of hint at a crosstalk between mitochondrial and protein homeostasis that is probably going on, and which is a very interesting area to study. So finally, we also measured cell death by this tunnel assay, where these green spots are, apoptotic nuclei, and again you see more cell death in the patient neurons. Also by this plate reader-based cytotoxicity assay, where autophagy inducers are protective. So in nutshell, we see that autophagy induction in our hands is cytoprotective in Wolfram patient-derived neurons, and therefore I kind of demonstrated that the same drug are effective in patient neurons of two diseases. So finally, the last part of my talk, I have mentioned that, autophagy is critical for cellular health, particularly for post-mitotic cells like neurons, because they can't divide. Therefore, they have to remove the cellular rubbish, and autophagy dysfunction leads to neuronal cell death. But you might ask the question, so what, who cares? So therefore, we are trying to address this question, that how loss of autophagy in human neurons mediates cell death. So this study was done by two of my PhD students. One is Elena, she's in the audience, and I'll speak more about Elena later in my talk. And another is Sun, in collaboration with Victor at Newcastle and Rudolph at the Whitehead Institute. And so what we did is we wanted to generate autophagy knockout human neurons. For that, we used human embryonic stem cell, which is one of the most physiologically relevant cell type in contrast to iPS cells. So using TALENs, we knocked out exon 3 of the essential autophagy gene ATG5. So what happens here is that if you knock out ATG5, this complex is critical for this phagophore formation. Knocking out ATG5, you do not have any phagophores, and therefore you do not have any autophagism. So you can see in multiple clones, we confirm knockout of ATG5. There is absolutely no LC32, as you can see by Immuno and Western, and there is accumulation of P62 because there is no autophagy going on. And we also use starvation, where you can see that you can induce autophagic flux in the wild type cells, but nothing happening in the knockout. So next, we differentiated these human yeast cells into neural precursors and neurons. We characterized by cell-specific markers. More characterization are published here, and we also confirmed loss of autophagy. You can see no ATG5, no LC32, massive accumulation of autophagy substrates. So next, our interest is cell death. So we are modeling by two different assays. One is cleaved caspase-3, another is this cytotoxicity. And here you can see that in two different autophagy knockout clones, there is significantly more cell death compared to the wild type. As we differentiate into neural precursors, we see small but significant increase in cell death. But this cell death is more pronounced after 4 weeks of neuronal differentiation. So you can see that autophagy knockout neurons are dying faster. So therefore, we confirmed the mouse data, that was published in 2006, that basal autophagy is important for neuronal health, whereas loss of autophagy is directly causing neuronal cell death. So next, the major question is how? So because of this rapid cell death, we hypothesized it could be due to a metabolic failure, and therefore we did metabolomics and identified NAD as one of the key drivers of mediating cell death. So what we found is that during loss of autophagy, there is hyperactivation of NAD-consuming enzymes that is reducing the total NAD pool, that is kind of affecting mitochondrial membrane potential and oxidative stress. And interestingly, this is also linked to protein homeostasis as well, and therefore leading to cell death. So I'm not going into the details, which is all published here, but just want to highlight that we confirmed the metabolomics data by additional biochemical assays that there is indeed depletion of oxidized and reduced form of NAD, and the oxidized form is predominantly reduced in the cytosol, whereas the reduced form is predominantly reduced in the mitochondria, therefore affecting this membrane potential. So then we also used various NAD boosters. These are NAD precursors, and we found that these NAD boosters can increase intracellular NAD levels and rescue cell death. And this was also associated with fixing mitochondrial bioenergetics, where you can see these NAD boosters are restoring mitochondrial bioenergetics in autophagy knockout neurons, as well as reducing agrosomes. Now this is very interesting because there is no autophagy. So how are these aggregates being removed? And therefore we also used chemical chaperone trehalose to show that these aggregates are being cleared, but probably this is due to fixing mitochondrial homeostasis as well. So finally, we also, in a parallel study with Victor at Newcastle, we showed this as an evolutionary conserved process in yeast, Drosophila, and in mouse cells, that loss of autophagy leads to reduction of NAD pool, and NAD boosting agents can improve cell viability and survival of the organism in autophagy knockout models. And finally, we wanted to show that in the context of disease, that we showed in a genetic autophagy knockout model that NAD is depleted, causing cell death. And now in a disease model that is associated with autophagy defect, we see a reduction in NAD pool, which we also recently published as in metabolomics. And using NAD booster can increase NAD levels and reduce cell death. And we see the same thing in wool from patient neurons as well, that there is a total reduction in NAD pool, and this was associated with autophagy defect as well. And NAD boosters, now we have included a new one called NRH, can significantly improve mitochondrial bioenergetics in all the Wolfram neurons. It can rescue the agrosome phenotype, cell death phenotype, as well as restore mitochondrial membrane potential, suggesting that this is cytoprotective in patient neurons. And finally, beyond 2D neuronal system, we are now developing 3D brain organoids. These are mini brains using the Lancaster protocol that was published before. And we see that as the autophagy knockout organoids are being developed, they are much smaller in size. And transcriptomics data suggests that there are problems with brain development as is featured here, and we are collaborating with Daniel here as well. So we are expecting to get the proteomics data on these cerebral organoids as well. So we also see that these organoids are associated with more cell death and as well as increased protein aggregation and DNA damage, suggesting that loss of autophagy in mini brain is likely causing neurodegenerative phenotype and various other problems, but we are in the process of characterizing. So finally, to summarize, there are many contributing factors to neurodegeneration. This is just naming a few, and we propose that impairment of autophagy is one of the major contributing factor, and we suggested NAD depletion as one of the causative factor, but of course there are other mechanisms, and these are some cover images that we got from the lab. This is autophagy knockout neurons showing cell death, and this is a plasticine model where this is a wild-type neuron with nice mitochondria, nucleus, and autophagosomes. This is autophagy knockout neuron, where the neuron is degenerating, fragmented nucleus, mitochondria, and so on. So I thank to all my lab members and my key collaborators. Here in Brazil, I'm collaborating with Edesio through our seed fund, Birmingham USP seed fund. With Daniel, we are collaborating as well through Birmingham Unicam seed fund. And I thank all my collaborators. And sorry, I just want to highlight that I'm also an advisor for NMN Bio. So Elena, who is speaking tomorrow, is my first PhD student, and she started this company. And the NMN that I have been using in my research is from her company. And we are happy to collaborate if anyone wants to use an NMN in their research. And just want to finally highlight, this is a variety of funding schemes that are available. So you can visit the website, you can just Google it, University of Birmingham Brazil Institute. Specifically for Sao Paulo, it's extremely relevant, but as you can see, there's UBBI is University of Birmingham Brazil Institute. There's seed fund. It's up to £15,000. It's British British pounds. It's match funding. Half and half comes from each university. There's also with USP. And then there is a FEPESP collaboration fund. I think this is a larger fund. And this is the FEPESP UKRI. This is more than about £1.5 million, British pounds. So this is eventually this is the target once you go through the ladder. But right now there's a Brazil visiting fellowship scheme. This is open to early career researchers, postdocs, and assistant and associate professors. And I'm kind of, Mary Elliston is the contact of the Brazil Institute. And again, you can contact me. I can try to help you with matchmaking to find, host labs in Birmingham. So thank you very much.
Well, congratulations, amazing talk. I have a question about your autophagy inducers, the chemical compounds. Actually, how do they bypass the genetic deficiency to activate autophagy? You know the mechanism.
Yeah, so in both the disease or in Niemann-Pick?
The first half.
The Niemann-Pick. So basically what we saw that the autophagy impairment is not absolute. So Niemann-Pick is kind of, the mutation in the protein is preventing this autophagosome-lysosome fusion. But I showed you a simplistic pathway, autophagosomes fusing with lysosomes. But in most cases, they first fuse with late endosomes to form an intermediate vesicle called amphysome. And these then fuse with lysosomes. Now, a small proportion of autophagosomes directly fuse with lysosome. So what we saw is that, triggering autophagy is causing newly synthesized autophagosomes to rapidly fuse with lysosomes and bypassing the amphysome step. And it's also important to understand that in majority of these diseases, the impairment is not 100%. So you can force stimulate it, but one factor must be considered is that lysosomal function should be okay. So if lysosomal function is screwed, then there's no point, rescuing it. Yeah.
Thank you.
Yeah.
Great talk. I'm wondering, because autophagy is very important for cell remodeling and cell differentiation. When you disrupt autophagy, do you see any problem with the differentiation of human embryonic stem cells? Do the cells develop normally or in neurons? Thank you.
So that's an excellent question. The neurons that we showed, are kind of immature neurons. So we just wanted to do the biology. We kind of reached 4 weeks of differentiation, but at that point you do not see action potential. So one of my collaborators tried to do action potential, after differentiating for 3-4 months. The knockout do not survive till then. You can get, wild type action, neurons doing action potential. Recently in a paper that we submitted on an organoid system, we see that the organoid development is totally bad in autophagy knockout. We are now doing brain organoids. Of course, we haven't characterized the markers and the brain layers. We are also doing single cell RNA-seq, for example, but I'm just showing you some phenotype that because of this massive cell death, there's likely that a lot of cellular populations won't be there. And it's this is kind of the genetic study kind of implicating that if there is autophagy defect in a disease, there's likely to be all these bad things happening. So the genetic studies are more concrete proofs of the importance of autophagy in a particular organ or a disease. Yeah.
Please, when you will start talking, please say your name first, okay? And where you come from.
Sanjar, my name is Rafael. I am a postdoctoral of ECB, and at the moment I have a project with mTOR, autophagy, and Alzheimer's disease. So I have 2 questions. The first question: the drugs. Do you know how much the drug can pass the blood-brain barrier, or rapamycin or not the drugs? How much can stay viable of the neurons? And the second question: what do you think about the defects of the blood-brain barrier can collaborate to the neuron disease?
Yeah, so this is a great question. So first of all, we have published quite a few reviews, and there are other reviews in the market as well, where you can see a list of autophagy inducers that are protective in the brain in mouse model of various neurodegenerative disease, including Alzheimer's. And one of the drug is carbamazepine. So I know for sure, there are papers out there that carbamazepine is clearing A beta, is improving cognitive, deficits in mouse model, right? Now, we have recently done a mini screen. I haven't shown the data. We identified 25 to 30 FDA-approved drugs that are known to cross the blood-brain barrier and are known to induce autophagy as well in other systems. So we took these 30 compounds and we tested in neurons to see what's the best one. And we kind of came up with 4 hits. Another criteria now we applied is what out of these, drugs that we selected based on blood-brain permeability and, autophagy induction, can they be used for pediatrics? Can they be used in children? And that is also, we applied this filter and we came. So there are lots of compounds of which I can advise, separately. Yeah, thanks.
Thank you very much.
Great talk, very interesting data. I have one question about lipids and autophagy, especially, how cholesterol, the accumulation of cholesterol translates in change in autophagy.
Yes, so, lipids, cholesterol are extremely important because you see autophagy encompasses several vesicle fusion events and membranes, have lipids, therefore. So, in Niemann-Pick, for example, cholesterol depletion strategy is common, with cyclodextrin. So but it has mixed results. You see, there is no treatment for Niemann-Pick. So in my study, I treated Niemann-Pick cells with cyclodextrin and checked autophagy. And I can see dose-dependent impairment in autophagy. As you increase the dose, there is again, already there is a block in autophagy. There's further defect in autophagy caused by cholesterol depletion. So therefore, yes, so when you are depleting intracellular stores, it is affecting vesicle fusion. And interestingly, what I didn't show that in Wolfram syndrome, it's not known to be a cholesterol storage disorder, but we used BODP and Philippine staining to measure lipids and cholesterol, and they're increased. And then you see the same feature in the Niemann-Pick as well. So I think in a scenario with autophagy defect, you will see an increase in lipids. Yeah. All right. Thank you.
Thanks, Alvin. Great talk. Danielle from Unicamp. So I was wondering, you said you have several chemical compounds that can activate autophagy, right? But then you also said that these are somehow specific to different cell types. Can we consider then that probably some of those are not directly aiming at autophagy, but something else, which in turn will, influence autophagy? Meaning that how can we say that whenever you use like a chemical compound, this is going straight to autophagy and activating it, or it's activating something else, given autophagy is something so central, that will activate autophagy? Well, does that matter actually, or maybe it doesn't matter? Maybe the thing is just acting over autophagy, right?
I think in the context of the biomedical importance of autophagy, this is a fantastic question. And the main limitation in the autophagy field is lack of a specific autophagy inducer that is acting on the machinery. When I mean the machinery, is the machinery involving ATG proteins that I did not describe. I mean, Oshumi got the Nobel Prize on autophagy for discovering these ATG genes. Now, apart from this, above the machinery, there's signaling pathways. So most of the drugs that I mentioned are hitting the signaling pathways that are, directing, the signals to the machinery that is stimulating the autophagy. Ideally, we need a specific compound that is hitting the machinery, and which is lacking right now. So right now, I think either you go for novel compounds, keep screening for it, or you try, for faster clinical translation, you use repurposed drugs, which are better tolerated, lesser side effects, and so on. But in terms of inhibitors, there has been some specific inhibitors such as ATG4 inhibitors and so on. So inhibitors, there are specific ones, but not inducers, unfortunately. Do you know when you disrupt autophagy, it triggers epilepsy, carbamazepine? Yeah. Yeah, yeah. So first of all, I don't know, and I'm not aware if the information is in the market. But I can suggest that like I collaborate with Tim Barrett. So he's currently doing a clinical trials with Wolfram children. Okay, and the results will be out soon. And these Wolfram children have epileptic episodes. Okay, and so interestingly, he's using valproate. So he's using valproate for a different purpose. But valproate also induces autophagy. So here we want to see what happens with the outcome, like control patients with valproate. Valproate is also an autophagy inducer, and I want to see. Still, we cannot say that autophagy is rescuing epilepsy because valproate does many other things. Yeah, so it's a great question, but I don't know. Thank you.
So carbamazepine is also used as a neuromodulator for many other mental disorders like bipolar disorder, etc. So it's and valproic acid as well. So there's a nice neuronal drugs.
Yes, but what we are seeing that these NAD boosters are amazing right now, and again I think Elena will be talking tomorrow. But I just want to say that NMN, it's a very competitive market, the NAD, and many of these NAD precursors are patented, means they're licensed, and you'd have to do a lot of stuff. Now NMN is not licensed; it's off patent, and we get clinical grade NMN from Elena, and that's something we are showing here. We have been collaborating with other partners, like we show that this NMN is rescuing zebrafish model of Wolfram syndrome. We are currently doing in collaboration, doing a testing in Huntington's mice about this, and the eventual idea is to take this nutritional supplement and autophagy inducers, repurposing drugs to the patients. So it's so these are emerging data that hopefully in a couple of years' time I will be able to show.
Okay, so I would like to invite the speakers of this morning session to the table so that we have a quick 10-minute panel discussion. Okay, so you can please come. Okay, so we can have 10 minutes. So, any questions about any of the talks of this morning, or would you like to ask questions across two different talks? Please say your name and where you come from.
Thank you. I am Mateus. I am from Edesius Lab here at the Heart Institute of the Faculty of Medicine. And my question was for Professor, Sankar. Yeah. I saw a recent study, but I couldn't find it on my phone due to my internet. But I saw a study in which they gave patients with mild cognitive impairment rapamycin, but at doses like 100 times lower than the doses that are used for immunosuppression. So the side effects were low. And they gave it for the patients for like 4 to 6 weeks. I don't remember the duration of treatment exactly. But what they found is that there was a cognitive, the progression of the cognitive impairment was lower than in the control group. And this improvement kept going when the treatment was stopped. So they evaluated the patients 1 year after, and they still kept getting better. So what do you think are the perspectives of using rapamycin in the clinic? And rapamycin is very lipophilic. Answer your question. So it crosses the blood-brain barrier quite easily. Thank you.
It's a very good study, right? I mean, of course, I haven't seen it, but looks very promising. So here I can suggest that we have to see the sample size, and the dosage, and the most important is whether it's inhibiting mTOR. Whatever dose, I think rapamycin at a small dose can also inhibit mTOR. So probably, again, we have to see the sample size, but definitely very promising. But logically, you don't want to use rapamycin because there are so many autophagy inducers in the market right now. I mean, why do you want to use rapamycin unless there is a specific reason for it, that it is better in inducing autophagy, or you want to do many things at once, by inhibiting mTOR. So still, in general, the research community believes that mTOR independent is more clinically desirable for lesser side effects than inducing rapamycin. There can be, some studies showing benefits, but there are converse studies as well, like rapamycin really deteriorated an ALS mice, okay? So it was really bad on an ALS mice. So there can be mixed studies, in the literature. So, yeah.
Some more questions?
Thank you. We have seen using rapamycin that some tissues, they do have a issue in getting rid of the rapamycin. For example, in adipose tissue, even if you use a very low dose, rapamycin accumulates in the tissue, and you do see a strong inhibition of mTOR in mTOR1 than other tissues that are able to get rid of rapamycin. So even though you are using a low dose at the long time with the accumulation, I think you'll be harming mTOR, and you're gonna get all the side effects of a chronic rapamycin inhibition.
Hi, amazing talks from everyone. My name is Cleo. I'm from Fiocruz, from Rio. And actually I work with neurodegenerative diseases. And in the genetics part, so I'm just curious about mitochondria and autophagy especially. And I wish to gain some insights. So what do you guys think about why these diseases, neurodegenerative diseases, they only start appearing in an older age, like when people are like in the 60s or something like that, and it's like autophagy is modulating something, or there's a lot of other mechanisms that may be involved in this process.
Yeah, I mean, you asked $1 million question. Nobody knows the answer. So, of course, there are hallmarks of aging, right? So mitochondria is an important factor. Now autophagy is also considered very important. NAD levels decline. That's also one of the features. I think, like with older age, like as you age, so there are studies in young versus aged mice where you can see autophagy decline, how that is affecting, tissue homeostasis and the organism function and everything. So yeah, I mean at certain age many things decline, and I mean because I work on autophagy, and based on strong genetic evidence in the literature, it's critical that the neurons must have good autophagy, because if autophagy decline, they can't get rid of bad mitochondria and other garbage, and there will be metabolic problems as well. So even I think there are some new reports of something called tunneling nanotubes. Like, there are some cell-to-cell connection, let's say, between neurons and microglia. And these are small projections that the cells are connecting. It's called tunneling nanotubes. And emerging research show that, neurons are more fragile than microglia. So when neurons are under stress, they get rid of the bad mitochondria. They send it through these tunneling nanotubes to the microglia. And the microglia sends good mitochondria to the neurons. It's like trying to help the neighbor, and these are, emerging studies. But yes, so I'm just giving you a very generic answer, so there's nothing specific. But the main question is selective neurodegeneration. Like different compartments of the brain are affected in different diseases, so we still don't know what is happening. And emerging studies are showing that even if there is autophagy impairment in a disease, different brain cell type can have differential impairment of autophagy to different extents. Some can tolerate, probably not having much of a detrimental effect of autophagy defect in such as microglia or astrocytes maybe, whereas certain neuronal populations are very sensitive and vulnerable. So still people don't know why selective neurodegeneration is happening in specific diseases.
Thank you.
To Julio, just trying to link the topics. I realized that in your electron microscopy pics, like clearly mitochondria is not just like modified in terms of morphology, but it seems damaged. Like the crista seems quite messy. Do you think like you could like perhaps rescue phenotype or improve this by adding like an AD booster or any other autophagy booster actually to improve your phenotype?
Yeah. You're right. Those mitochondria are just like giant swollen mitochondria, which is harder to remove as well because the autophagy you need. I mean, the size matters. So if you have huge, especially in skeletal muscle, that it's very packed. I mean, so that's something that we're trying to. If you rescue the mitochondrial morphology, is it sufficient to get rid of those damage? Because I haven't mentioned, but in those animals, the autophagy flux is also disrupted. So that's another reason we accumulate those huge mitochondria. But that's a good point. You don't know if you kind of decrease the threshold of the damage and then you can connect both systems. Again, that's a good question. Yeah, probably yes.
Yeah. So if I may add, I think normally, there's a specific size of autophagisms, let's say 0.5 microns. So when there is infection, there's a selective autophagy process called xenophagy, where bacteria like mycobacteria, it's a big structure, invades. So they can capture, the autophagisms can capture them and they become large, like 1.5 or 2 microns, So maybe, in your scenario, there might be, some large autophagisms, it's basically membrane that is wrapping around the material that needs to be removed. So if you do any live cell or even fixed just immuno, or even if you do immunogold, electron, microscopy, maybe, you can get some answers whether these are indeed, whether autophagy is trying to get rid of that.
Yeah.
Someone else?
Hello, hi. Hola, hello. It's Professor Celia. Professor, in your mitochondrial disease studies, did you try to treat the cells or the patients with any NID boosters as someone target? Like some kind of improvement and supplementation helping the mitochondria to produce more energy?
I didn't, but there are some groups trying to do that. So I don't know the results yet, but they are trying to.
Okay, now so we're past our time. I thank you. I thank all the speakers of the morning, and I thank you for coming here. And let's reunite at a little bit before 2 p.m. today. Okay, thank you very much.
Hello, everyone. I'm João Paulo. I'm going to be the chairperson this afternoon. And now it's a pleasure to introduce Professor Adécio Cunha Neto. He's an associate professor of clinical medicine and researcher at the Heart Institute of the University of São Paulo Medical School. He holds a PhD in immunology from USP and was a visiting researcher at Stanford, Harvard, and institutions in Italy and France. He leads the Laboratory of Clinical Immunology and Allergy and coordinates USP's graduate program in allergy and immunopathology. His research focuses on molecular immunology with an emphasis on chronic Chagas cardiomyopathy, vaccine development, and biomarkers for infectious, autoimmune, and allergic diseases. Please, everyone, welcome Professor Adécio Cunha Neto.
Thank you, João, for the introduction. So we had Celia's presentation about human disease. So it was very nice because she introduced lots of things about mitochondria for us. So I'm going to be talking today about the pathophysiology of Chagas disease cardiomyopathy, and specifically the cytokine-induced mitochondrial dysfunction, which I'll try to convince you is a new paradigm and therapeutic target for the disease. So Chagas disease is caused by the protozoan parasite Trypanosoma cruzi. Yeah, it's endemic in Latin America, but due to migration, there are large populations of Chagasic patients in Europe and the US, especially, or it's more than 400,000. So the people there, they're starting to do heart transplants on patients with Chagas because there are so many. So the transmission forms are basically the vector, insect vector, which bites and then leaves the poop with the infecting parasites. It can also be transmitted congenitally, orally, through blood or organ donation. So the triple subocclusive life cycle in the mammalian host. So we have the triple mastigotes that those slender forms I showed in the first slide, which will enter a cell. Then it will change form for this A mastigote. It's a round cell without a flagellum. And this is the replicating form. So it will be replicating a lot in the cell, and it will then transform again into triple mastigotes, infected triple mastigotes, and then this cell will burst and release all these triple mastigotes, which will be infecting the other cells. So the natural history of Chagas disease, we have an acute infection for the first weeks. We have high parasitism. Then this parasitism is really controlled to very low levels due to the innate and adaptive immune responses. In a long time, we'll have 30% of patients developing chronic Chagas disease cardiomyopathy. 10%, 1/3 of those will develop especially severe cardiomyopathy, the severe Chagas cardiomyopathy, which is has worse prognosis than other cardiomyopathies. And 10% will develop digestive disease, but we're not going to talk much about digestive now. So this is what a heart from a patient who Chagas disease looks like when it's end stage. This heart was taken out of a patient during transplantation. And if we can look here, this is HE, this is precocidus for fibrosis. We can see here that in Chagas cardiomyopathy, we have increased inflammation, much more than dilated cardiomyopathy, and controls, of course. And there's also higher collagen deposition in fibrosis in Chagas as compared to dilated cardiomyopathy. So in Chagas, the myocarditis intensity and frequency, it's associated with Chagas heart damage. So if we get heart biopsies from people who have no heart disease, this was done 30 years ago, so the ethics committees were not concerned about getting these kind of samples. So we have a very small percentage of people with myocarditis. This increases a lot in people who already have some initial heart symptoms, and it's really very high. It's essentially all of the patients will show intense myocarditis during severe cardiomyopathy. So for severe cardiomyopathy, as I mentioned, it has a lower survival, it's red here, as compared to non-inflammatory cardiomyopathies, either ischemic or idiopathic. And we know now that standard heart failure treatment is not as efficacious for Chagas as for the other etiologies. So what we know there is that Chagas has more than the dilated cardiomyopathy. There's more inflammation, more fibrosis, and there is a more severe arrhythmia. So the open questions are, what is the pathogenic basis of disease progression? Why do 30% of infected people develop Chagas cardiomyopathy? Why do some patients develop the severe form? And why is it that CCC has a worse prognosis than other cardiomyopathies? And we will be looking for the translational potential of the information about the pathogenesis. So when T-Cruzi infects the host, it will trigger an innate response with upregulation of cytokines like IL-12 and lutein, which will drive the differentiation of T cells that produce interferon gamma and TNF alpha. So this is really important because this is going to increase the ability of macrophages to destroy the intracellular parasite. But at on the other side, it's like a double-edged sword because if you have high levels of interferon gamma and TNF alpha for a long time, you may get tissue damage. So these are plasma levels of interferon gamma and TNF alpha. So here is interferon, here is TNF. So we see that already in the indeterminate phase, you have an increased levels of TNF alpha and interferon gamma. But then when you get to see the patients with cardiomyopathy, then they have really much more higher levels of interferon gamma and TNF in TNF alpha in the peripheral blood. And you can also see that in if you check for cells producing interferon gamma in the blood. So what happens in the heart? So we've been studying heart-infiltrating T cells to elucidate the pathogenic process. We've been able to show that there are CD4 helper T cells that are capable of cross-reactively recognizing trypanosoma cruzi antigens and cardiac myosin, which is the major heart protein. We could also identify T cells that recognize trypanosoma cruzi antigens without any cross-reactivity. So when we look at the cytokine profile of these cells, once they are activated by antigen, we see that this is immunohistochemistry staining for interferon gamma. So we see there are many mononuclear cells there, and they are all stained with interferon gamma they produce it. And this is a study we performed where we got biopsy samples, and we obtained the T cells from each biopsy sample, and we stimulated them and tested for cytokine production. And we saw that for all of the samples that we tested, we had interferon gamma TNF alpha production. So the next step, we wanted to unravel the pathogenesis. We tried to do a transcriptomic and proteomic profiling of the myocardium of these patients. So we used Agilent microarray and DIGE and LCMS approaches for proteome. So we found a number of 1,500 differential expressed genes and 404 differential expressed proteins. And this was led essentially by Priscilla with also involvement of Laurie and Ludmila. So when we looked at the pathways, we saw that the key pathophysiological processes we had a large number of differentially expressed genes, so inflammation, interferon gamma response, fibrosis, matrix, hypertrophy, and then we had oxidative stress and mitochondrial genes. So the next question we asked is what's the role of interferon and TNF alpha in changing the transcriptional profile of the heart? So we use ingenuity pathways analysis, and we did the upstream regulator analysis. So this will indicate to us which are the most likely ligands that will induce the transcriptional profile. So the top upstream regulator was indeed interferon gamma, and the second one was TNF alpha. So when we did GSEE analysis for pathways, we saw that the induced pathways, they were all related to inflammation, interferon gamma signaling, but when we looked at the repressed pathways, we saw several repressed processes which were related to mitochondrial function and energy metabolism, such as mitophagy, mitochondrial translation, electron transfer chain. So, and then we did a tox function analysis for pathways, and we could see that we already had the major processes there, and in some cases, so the red bar here is Chagas, blue is idiopathic, and green is ischemic. So for some of these, there are several mitochondrial processes here, and for some of them, there was a really much higher match. For decreased transmembrane potential of mitochondria was much higher in Chagas as compared to the other cardiomyopathies. And then we have oxidative stress and NRF2. So, in this proteome, we're able to assess many things, but there was a prominent change in energy metabolism proteins. So we saw reduced expression of beta oxidation, Krebs cycle, respiratory chain, ATP synthase, UCP2, and creatine kinase shuttle proteins. And they were especially severe. The depletion was especially severe in Chagas for beta oxidation, ATP synthase, and creatine kinase. So would expect that this would make would bring a situation where ATP production would be reduced. in Chagas hearts. So this is what happened with beta oxidation. So in green, you have the decrease in enzymes. So there are 6 beta oxidation enzymes that are decreased in Chagas, as opposed to 1 or 2 in idiopathic dilated and dilated cardiomyopathy. So and this is a Western blot showing just the very long chain SQA dehydrogenase, which is here. We validated this in Western blots. So we also know that mitochondrial ultrastructure is disordered in Chagas hearts. So we can see some edema of the lamellae, size and shape irregularity, and mitochondrial disruption in some cases. So we know now for a long time that mitochondrial bioenergetic efficiency is reduced in heart failure as a whole. So we have a reduced mitochondrial membrane potential or proton gradient. We have an increased electron leak. So we have in heart failure we'll have dysfunctional mitochondria. So what happens when we compare energy metabolism in the hearts of patients with Chagas disease as compared to idiopathic dilated cardiomyopathy. So this study was done comparing a control group, and we're looking here at high energy phosphates. This was done with phosphorus spectroscopy in living patients, so it's in vivo. So we know that already in Chagas disease patients without heart failure, there's a reduction in high energy phosphates. And this reduction is even more important in Chagas patients with important heart failure. So, and when you compare patients with the same heart failure class from Chagas disease and from idiopathic dilated cardiomyopathy, we see that in Chagas hearts, you have even less than that. And the level of high energy phosphates here is really in advanced heart failure or of non-Chagasic etiology is similar to Chagas disease without heart failure. So there's an energy metabolism impairment that starts before heart failure in Chagas heart disease. So for the first summary, when we looked at the transcriptomics, proteomics, and some metabolomics, which was done in another group, so we see increased inflammatory cytokines, and interferon gamma-driven transcriptional signature, chemokine profile that we are attracting cells that produce interferon gamma. I'll show you this later. And we saw reduced multiple pathways of mitochondrial energy metabolism, quality control were reduced, and we had increased oxidative stress. Some of these I didn't show today. So for the CCC hearts in vivo, as I mentioned, the depletion of high energy phosphates. So now that we had, there's intense interferon gamma-driven inflammation, and there is a mitochondrial dysfunction. Is it possible that the interferon gamma is driving this mitochondrial dysfunction? So that was the question here. So as a model, we used a model system. We could try to compare myocardium, Chagas myocardium, and A16, an in vitro model using A16 cardiomyocytes treated with interferon gamma and TNF alpha. So this is essentially João Nunes' work together with Pauline, with the 2 Paulines in France. So and this is an image of a mitochondrial network that we got with the Revity operator system that we just got. So we observed that there is a reduced mitochondrial DNA in Chagas cardiomyopathy in hearts, and this is also recapitulated in cytokine-treated IC16 cardiomyocytes. There is an increased nitrite NO production, and this is also increased in cytokine-treated cardiomyocytes. Finally, we have an increased oxygen-reactive species here by the DCFDA stain, and we also have a reduction in ATP production on treated cells. And we did seahorse analysis of these cells. So here I'm showing total ATP, glyco, and mito ATP production. So we saw the cytokine treatment reduces all of the forms of ATP production. We also saw that cytokine treatment increases respiration and increases proton leak. So we might be seeing here a situation of mitochondrial uncoupling. Further, when you analyze substrates for energy production, we found that cytokine-treated cells, they had a reduced fatty acid dependency. So this is in line with this reduced fatty acid beta oxidation we saw in the tissue. So this here is not going to be easy to see, but you see here there's a lot of bright red, which is TMRM, shows polarized mitochondria. And these cytokines are isolated and together. You see that here it's much dimmer. So there is these mitochondria are less polarized. So if you quantify that, we have this kind of graph. When João asked the question whether mitochondria, whether large mitochondria or mitochondrial networks, or mitochondria larger than 10 micrometers in area, whether they also show this cytokine-induced dysfunction, reduction of MMP, and you saw that even with longer mitochondria, you saw reduced mitochondrial membrane potential. So then we studied the pathways that were important for the cytokine-induced reduction of membrane potential. And we found that STAT1 inhibitors and FKB inhibitors, inducible nitric oxide synthase inhibitors, and P38 MAP kinase inhibitors were all able to increase the mitochondrial membrane potential to baseline concentration situations. So we also saw that there were agonists or reducers of AMPK and NRF2 and sirtuin-1, they also increase MMP in these cytokine-treated cardiomyocytes. So we could rescue that with this kind of chemicals. So then we did a proteomic analysis of the cytokine-treated cardiomyocytes to look at the differentially expressed proteins and compare that with the proteomic profile in Chagas heart tissue. So we had an overlap of around 50 proteins. 35 of these were concordant. They were up or down regulated in both sides. And 10 of them were mitochondrial. And we saw a reduced expression of fatty acid oxidation, TCA, electrotransfer chain, and antioxidant response proteins, and increased proteins related to glycolysis, mitochondrial biogenesis, and apoptosis. So if we put this all together, we have this summary slide is showing the effects of cytokine treatment in the cardiomyocytes, and this is the findings in Chagas myocardium. So you can see that in most cases, we have changes in cytokine-induced treated cardiomyocytes, which are similar to the findings in Chagas myocardium. And if you do pathways analysis, we are going to see increase interferon gamma signaling, but reduce mitochondrial, so increase mitochondrial dysfunction, decrease the transmembrane potential, reduce electron transfers, TCA cycle, fatty acid beta oxidation, and with an increase in glycolysis. So for this, so when we put together all of this, we concluded that cytokine-induced mitochondrial dysfunction may be a key pathophysiological mechanism in Chagas cardiomyopathy. So we've wanted to study further what happened to these cells, to these cardiomyocytes, after we treated them with interferon gamma and TNF-alpha. So there's they induce mitochondrial dysfunction, but will they change the transcriptional profile of these cells? So what we did is we did real-time PCR analysis of treated and non-treated cells, and we saw that there was an upregulation of expression of inflammatory chemokines, especially the ones that are able to induce migration of inflammatory T cells. We could also see increases in IL-1 beta, IL-6, IL-12, and we could also see increases in interferon, in the type 1 interferon response, interferon beta, and the interferon-inducible genes. What was interesting to show was that when we treated the AC16 and we had an increased levels of CCL5, CXCL9, CXCL10, chemokines that attract inflammatory T cells. So this is an immunofluorescence showing that we see both CCL5 and CXCL9 here, positive cells. And so then this is the real-time PCR showing that indeed we also have increased CCL5, CXCL4, CXCL9, and CXCL10 in heart tissue. And these are cytokines that can increase inflammatory infiltration. Actually, we did then a correlation analysis between the level relative expression of CXCL9 and the myocarditis scores as the degree of inflammation. And we saw there was a correlation with many chemokines, but with CXL9, this was the most significant one. So I'm going to talk here a bit about common genetic variants versus susceptibility to Chagas cardiomyopathy development. This is going to be described in much more detail by Christophe, who is going to present the following presentation. So the finding that only a proportion of patients infected by T-Cruzi will develop Chagas cardiomyopathy in the familial aggregation of Chagas disease cardiomyopathy cases like this. When you go to an endemic area that everybody is exposed, many people are infected, there are some families where there are several cases of cardiomyopathy and some other families where there are not so much, even though they're all infected. So we started to look here at genetic polymorphism in CXCL9, this chemokine that's increased in Chagas hearts and in cytokine in treated cells. So what we saw is that we picked up a polymorphism that was associated with inflammatory disease in other situation, and we saw that the A allele of this polymorphism it was associated with increased risk for severe Chagas cardiomyopathy. So then we compared. We had all the heart biopsies here, and we genotyped the heart biopsies, and they are separated here as to whether they have an A allele or do not have A allele. So the biopsies with the A allele had much increased CXCL9, so in the blue here, as compared to the non-A carrying patient with Chagas heart disease. So having the CXCL9 allele, increased allele, that was associated with increased risk for severe Chagas, was also associated with increased production of CXCL9. And then we looked at the risk allele, and we started to try to look whether it was associated with intensity of myocarditis. And again, in biopsies which had the A allele, we had a much higher inflammation score than in biopsies that did not have the A allele. So as far as a overarching thing, we believe that inflammatory cytokines will increase, for instance, in FKB. This will lead to mitochondrial damage through inducible nitric oxide and peroxide nitrite production, leading to mitochondrial dysfunction, energy deficit, heart failure, and Chagas cardiomyopathy. So as a result of this cytokine exposure, apart from the cytokine damage, you can have an increase in inflammatory chemokines that recruit macrophages and inflammatory T cells to the heart. So this is a positive feedback loop that may be operating in Chagas heart disease. So with all that, we wanted to propose a new paradigm that cytokine-induced mitochondrial dysfunction would be a central culprit in Chagas cardiomyopathy. And then the next question is, can we mitigate the cytokine-induced mitochondrial dysfunction to treat Chagas cardiomyopathy? So what we did, this basically the PhD project of Vinicius Roda was here. So we assembled a high-throughput screening assay with 384 plates to screen 2,300 different FDA-approved or nutritional supplements that are used currently on a system where we had the cardiomyocytes in the presence of cytokines. So we were looking for the compounds that were able to increase ATP production in the presence of cytokines. So we had many hits here, and we had a series of validation steps where we repeated the assays with several replicates several times. So we titrated the drugs that give us a signal to see which was the minimum concentration where they had that effect of increasing ATP production, and we also found the concentrations where there was no cytotoxicity. So the Vinicius was aided again by João, by Luis, by Rafael, who is not here anymore, and by the French team, Alizé, Pauline, and Elia. So at the end, what we have now, we have a handful of drugs which consistently ameliorate cytokine-induced mitochondrial dysfunction. And I'm going to just to show a bit of this. So we had cytoprotective effect salt. We had a glycoside with antioxidant and anti-inflammatory effects. We have a non-steroidal anti-inflammatory drug. We have an angiotensin receptor blocker, a methyl donor with anti-inflammatory effects, an amino acid with anti-inflammatory effects, and a cyclic disulfide with antioxidant properties. So what was a common feature to all of these drugs, and so at least 7 out of the 9 are drugs that are really repurposed. They are used clinically for other diseases, including angiotensin receptor blocker and ACE inhibitors. So, and we investigated whether that was a single molecule event or whether we had a class effect. So we tested several angiotensin receptor blockers, captopril and the other ones, and we tested valsartan and other angiotensin receptor blockers, and we saw that was a class effect, drug class effect. So the mechanisms that are described for all of these compounds include inhibition of NF-kappa B signaling and mitochondrial protection. So what we are now doing, will be next step for drug target validation is using an in vivo model with mice that are transgenic for high expression of interferon gamma in the liver. So these mice, they develop hepatitis, but they develop they have high levels of interferon gamma in the plasma, and they develop an inflammatory cardiomyopathy. So the design here would be to treat these animals before they get to the age with severe disease, and look for echocardiography and other biochemical, immunological evidence of inflammation and mitochondrial dysfunction in the heart. So, and the other thing, I was just last week was contacted by a cardiologist at the core. I'm an immunologist. I'm not a metabolism person. We started to do metabolism because the data started to point to that direction. So a cardiologist at the core approached me to say, we could, there's a good coenzyme Q10 company here which provides a highly bioavailable coenzyme Q10. Perhaps we could do a test with Chagas disease and assess VO2 or the things. Say, great, but let's coenzyme Q10 and was not one of the hits here, right? But so I'll be showing him this presentation in more detail next week or the other, and we may be able to, since these drugs most of them are really already used in clinics, we may be able to do a straight clinical trial or some kind of observational study straight from the in vitro data that we have here. So we believe that apart from looking for a treatment, possible treatment for Chagas cardiomyopathy, which is a very regional, local problem, if we get a treatment for that, for cytokine-induced mitochondrial dysfunction, we can also target and treat other inflammatory heart conditions like cardiac aging, where there's an accumulation of interferon gamma producing cells, inflammatory cardiomyopathies, viral myocarditis, and cancer chemotherapy cardiotoxicity. This is the last slide. Oh, yes. So I'd like to acknowledge the people who provided or produced most of this data. So João Paulo spearheaded the studies for more than 5 years in our lab. And we have Rafael Almeida, João is here, Deborah Levy, Deborah, I don't have the picture of Deborah here, Andrea Kuramoto here, Luis Felipe, Claudia, Vinicius Roda here, and Guilherme, and George Caliucci, who is the head of the department. So we have other collaboration with people in other places in the core, and we have a strong collaboration with Christophe Chevillat in CERN. Most of the data that we showed today was done in this collaboration, and this is the INSERM team in Marseille, headed by Christophe Chevillat. So thank you for your patience, and I will open for questions.
Okay, thank you, Adesio, for your presentation. It was really nice. Someone has question for Adesio?
Thank you. Very, very nice presentation. I think you made a very good job in proving your points, and I have no doubts about what you're postulating there. But I have some curiosities. Firstly, I think, I don't know if you or somebody else ever looked at the adipose tissue that surrounds the pericardium, because interferon could be coming from these cells. And we also find that, for instance, not in tumor, so it's a totally different thing, but the peritumoral adipose tissue is really secreting loads and loads of interferon, and it leads or not to cachexia, depending on the specific aspects. This is one thing. And the second thing is that actually, then we should talk later, but you mentioned some amino acids with metabolic or anti-inflammatory properties. And I would be very interested to have a look at a metabolite of leucine, HMB, and if you consider looking at it as well, because maybe it would aggregate, it would join the two properties that you're willing to have a look at. Thank you.
Great. Of course, you're more than welcome to come to us, and we can test your drug. So regarding adipose tissue, the beginning of the work said, no, you got explants from transplanted hearts. The first thing we did was to chop off the fat and throw it away, right? So the data are really about left ventricular free wall. But now it's true that even in heart disease, it's been shown that pericardial adipose tissue really is inflamed and can modulate the inflammation in the cells. The only thing about that is that when we get inflammation not only in the subpericardial region, but also in the middle and the subendocardial as well. But this is certainly an issue to be looked at in Chagas.
It's a curiosity and ignorance as well. Do obese people have more effects in Chagas disease, more symptoms and more complications?
Well, Chagas disease is socially related, so it's a disease of poor people in poor rural endemic areas. So historically, these people there were not too many fat people in this population, but nowadays with the obesity epidemics, it's possible that there has some change, but there has been no study in Chagas patients taking into account that. If you in animal models of infection, if you do high fat diet, for instance, you accelerate disease.
Any more questions?
Well done, Adesio. It's a great talk. I'm interesting about your screening. So you did a screening based on phenotype, right? And then you identify a couple of compounds that have a phenotypic effect. I would say a medicinal chemist will ask you, what about the mechanism of action of these compounds? And I know that you're moving to animal studies. So how are you going to select the compounds? Are you going to test all of them, or the ones that you know the mechanism of action and that you can actually generate more knowledge about a repositioning this drug for treating Chagas disease?
Yes, great question. So when I just mentioned literature findings about these compounds with the reported findings, but now we intend to study the mechanisms of this amelioration of ATP production, looking using an F-kappa B translocation to the nucleus as one of the studies, and more refined mitochondrial dysfunction analysis. So this is going to be done, and we want to, so when we move to the in vivo model, we want to have at most 5. So we're really looking at the best candidates now.
Nice.
My question is more about to hear your opinion, because I mean, your data is beautiful, but and I know that you are focused on improve cardiomyocyte health to treat Chagas, but heart, like any other tissue in our body, is very complex. We have other cells in our body, and you show us like beautiful pictures from patients, and they're like tissue fibrosis. What I'm thinking is that, like, even though you improve cardiomyocyte function, but you still have activation of fibroblast and tissue fibrosis, do you think that is enough to improve cardiomyocyte function in order to improve heart function of these patients? Should we like target fibroblast as well?
Sure. No, this is a great question, and it's actually, this is the subject of our next grant proposal. So we want to do spatial transcriptomics in the heart to exactly to look at the derangements in other non-cardiomyocyte populations like fibroblasts, immune cells, monocytes, and as you'll see, we have models of iPSC-derived cardiomyocytes, and we're going. It's in our plan to differentiate cardiac fibroblasts and monocytes from the patients, from Chagas patients, to be able to ask this question.
Mm-hmm.
Right. So it's really what we're focusing. In the
Super
future.
cool. Thank you.
We still have one more time for one question.
May I add comment on your answer? It's really, we can work on cardiomyocyte, and as you said, there is also fibrosis. We did the screening, and as Vinicius is going to do, he's looking for transgenic mice. But we have another system to test the drugs. It's Drosophila. Because when you are using Drosophila, if you are doing a high sugar diet to this Drosophila, after 10 days, they will develop like a cardiomyopathy with arrhythmia. And if you push the system up to 20 days, they will develop fibrosis. So by this way, we can test the drugs also on this live model, and we will have information about these drugs on arrhythmia and fibrosis. So it's another way to test the drugs. And of course, we will test the drug with Professor Edesio, each selected drug alone, and we will test this drug also in cocktail. And of course, when we are going to test cocktail, we are going to select some drugs that will target different pathways, not the same pathways.
Okay, so we move on for the next speaker. Thank you, Edesio, for your presentation. So the next speaker is Professor Christophe Cheviat. He earned his PhD in 1994 in Marseille, focusing on the genomics of Charcot-Marie-Tooth syndrome. after a postdoctoral fellowship in San Diego studying antibody gene organization, he returned to Marseille in 1997 to lead research on the genetics of parasitic diseases. Since 2010, he has focused on Chagas disease in collaboration with Professor Adesio, aiming to uncover disease mechanisms, biomarkers, and new treatments. Please welcome Professor Christophe.
Thank you, Jean. Thank you, Jean, for this introduction, and I would like to thank the organizer. So I'm going to start with this slide that summarizes the work that we've done during the last 15 years. So I had a chance to start a collaboration with Professor Adesio in 2010 on a beach of Santos. And 15 years later, we are still working together. And he's still not mad after me. So, but I feel very lucky because with Professor Adesio, I have a strong collaborator. And I can say today that it's one of my best friends, and I consider Adesio as my brother. Savan said he came 7 times to Brazil. You said that's what you said. Well, I came approximately 20 times since 2010, and I still not speak Portuguese. And I just realized that at the lunch times that each time I'm coming to São Paulo, I forgot how strong are the caipirinha to São Paulo. And when you drink a caipirinha at lunchtime, you are in trouble the afternoon like I am. So Professor Adesio asked me to focus on the genetic of the Chagas disease today. So I just want to remind you that Chagas disease is probably the worst cardiomyopathy disease, because after 2 years of chronicity, 60% of patients pass their way, and after 7 years, only 20% survive. You can detect the parasite only during the acute phase, and as the pathogenic process is mainly unknown, there is no treatment so far. So we have developed a huge program to decipher this pathogenic process, identify new biomarkers, and identify new treatments. So we start to do omics approaches, proteomics, epigenomic, genetic study, methylation analysis, metabolic, and transcriptomic. And we move now from single omics to multi-omics. And soon we will start omic approaches on purified nuclei of cardiomyocyte, and we will do spatial transcriptomic associated to imaging. So do we have an in vitro model for Chagas disease? And Adesio talk a lot about it. That's the AC16. So when you are asymptomatic, you have an immune balance that is relatively equilibrated, but as soon you do a CCC cardiomyopathy, you have a strong Th1 response. And you saw this slide, so you have a yin-yang effect of the interferon gamma, which may have a protective effect, but a deleterious effect also. Okay, and when you stimulate the AC16, you have an increase of the oxidative stress, a decrease of ATP, and you have a low mitochondrial DNA copy number. And as Adesio described previously, we have a concordance between the result obtained on AC16 and on patient. So, do we have a genetic compound in Chagas disease? So, the first things to do when you are doing genetic analysis is to do a case control study. So, you have your true group, your cases, your control. All the individuals are unrelated, and you test some polymorphism. If a polymorphism is more frequent in the cases, it's a susceptibility variant. And if the variant is more frequent in control, is a protective one. And you can see here all the list of genes that has been associated to Chagas disease. And 50% of them, written in green, has been identified by our consortium. But it's just case control studies. So the other way to do the genetic analysis is to perform a GWAS analysis. It's like a case control studies, but you are testing million and million of SNP. You will have to do association study, and you will have to do linkage disequilibrium analysis. And to perform this linkage disequilibrium analysis, you have to use a reference population. And people who are doing reference population, they are using generally the information coming from 1,000 genome. And in the 1,000 genomes, there is no reference population for South America. So it's a big problem. So 3 GWAS has been done so far. The first one is here, and it has been done by Professor Edesio. It gives 6 suggested loci that seems to be relevant for the disease. Since Javier Martin did in Spain did a new GWAS on a limited number of subjects, and he got only 1 significant locus. And Esther Sabino with Alexander Pereira here in São Paulo did the third one with a huge number of patients, and they identify only 1 locus. So we have a new GWAS ongoing with more than 2,000 people. We have some asymptomatic and a huge number of CCC patients. Some are moderate, some are severe, but we have also some patients with pacemaker or defibrillator. So it's a new phenotype. So what are we going to do differently from the GWAS? That's our power will be the reference population. It's a Brazilian population of 1,100 individuals, and their whole genome has been sequenced. So by this way, we are thinking that we are going to do a more appropriate reference population for the linkage disequilibrium. And we have now a way to test epistaxy analysis or functional test at large scale. That's the same. So the third approach that we are developing, and we got some significant result, is exome sequencing on multi-case nuclear families. So we did exome sequencing for all these individuals, and we were looking for coding variants that are shared by the cases, but not found in the asymptomatic subject from the same families. And for these families where we do not have asymptomatic siblings, we compare to this group of 14 unrelated asymptomatic patients. So we did the exome sequencing. We did the exome sequencing. We select the variant with a mean allele frequency under 1% just to get the rare variant. We select the non-synonymous exonic variant or splicing variant. We look for the pathogenic score of this variant, and we select the more pathogenic. We select the variants that are located in a conserved region among the evolutionary. And we look for the pathway of the gene that has been selected. And we identify 22 variants in 20 genes. And here you have the list, and all these variants are associated to mitochondria or inflammation. And in each family, you have at least 1 variant associated to mitochondria or inflammation. And it's really important because when you have a mitochondria disease, it can lead to multiple phenotypes, as Celia described this morning. So that's the case that we can get in Chagas disease. And of course, we have also the immune response. Okay. So our hypothesis is when you are just the mitochondrial variant, you will have some subclinical dysfunction. If you are wild type and you have the strong interferon Chagas disease with a strong interferon gamma response, you will have also, but significant, but also some limited dysfunction. But as soon you have the strong interferon gamma response plus the mitochondrial variant, you will have a strong dysfunction that will lead to severe disease. So we select a couple of these variants for functional study. We select one in DHODH. It's a complete loss of function. It's an heterous variant located in the exon 3 that lead to a change of one amino acid. And this has been this amino acid has been localized at the catalytic site of the DHODH. We select a variant in UMPS here, involved in the pyrimidine biosynthesis. It's also a non-synonymous variant that change an amino acid. And the third variant that we select is in RPU-SD3, involved in the pseudouridine of pyridine. And it's a variant located in exon 8, which induces a stop codon. And when you have a modification of the pseudouridine process, you have instability of the RNA with a decrease of mitochondria translation, a decrease of OXPHOS, and a decrease of cell viability. So all these three variants has been characterized, and I just want to repeat, this is a rare variant, pathogenic, localized in the coding region. So for example, if we are looking on purified mitochondria, for the DHODH variant, we have a decrease of the activity of the DHODH enzyme, and we have a lower concentration of orotat, that is the final product of this DHODH enzyme. And as you can see, we generate some CRISPR clone with a wild type phenotype or carrying the mutation of interest. For the UMPS variant, we also set up some CRISPR clone. And as you can see, when we are looking for UMP, we have a lower concentration of UMP on the clone carrying the mutation under without the interferon gamma stimulation. And for RPU-SD3, we got also some CRISPR. We did some Western blot to test all the complex of the respiratory channel, and we can see that the variant is affecting the complex 1, the complex 3, and the complex 4. So we try to push forward in this functional analysis. And we did some QRT PCR to double check the expression of this gene. And I can tell you, when we decide to do this experiment, we do not expect to get some change because the variants are located in the exon. And here you have all the variants for all the transcript for the DHODH, coding for different proteins. You have the exon 3 here, where the polymorphism is located. And when we did the qPCR, as soon as we test the heterozygote variant, we have a strong decrease of the expression of DHODH. So we were really surprised. But we push forward, and this is the exon 3. And here, that's all the transcription vectors that bind to this exome. And we push and we analyze multiple databases. And in fact, this exon 3 of DHODH is considered as an enhancer region. So by this way, our mutation is affecting the sequence of the DHODH protein, but it's changing the binding of transcription factor that will modify the expression of the surrounding gene. So we are doing actually some experiment to confirm this enhancer property, because it will be the first polymorphism associated to Chagas disease that is located in a coding region and that affect an enhancer region. And for the two other variants, we do not have significant change in expression. So we push forward on the characterization of this variant, and for most of them, as soon as we are doing the stimulation, we have a decrease of ATP production here for the DHODH, UMPS, but for RPU-SG3, when you have the heterozygote CRISPR clone, you do the stimulation, you have an increase of ATP. So, in the disease, we have the Th1 response, the NF-kappa pathway, and as shown at the show, there is multiple cytokines that has been differentially expressed between the AC16 with or without the stimulation. So the question that we ask, do the variants that we are characterizing modify this cytokine profile? And the UMPS variant and the RPU-SG3 do not modify this pattern. But as you can see, when we compare the DHODH CRISPR clone, CC or CT, we still have an exacerbation of production of multiple cytokine. And when we are looking for inhibitors for inflammatory chemokine, we are killing this overexpression detecting on the CRISPR clone. And for the cytokine associated to the Th1 response, we also have an effect of the tested inhibitor, and all of that has been done by Joao. Yeah, that's the same. I was thinking I did that. So now we look for the source of ATP. So we use two different inhibitors, etomoxil that blocks the fatty acid oxidation, and 2DG that will block the glycolysis. And as you can see, in our DHODH CRISPR clone, the main source of ATP is the glycolysis. When you are blocking the fatty acid oxidation, you do not change the production of ATP. And that's the same for the CRISPR clone for UMPS and RPU-SG3. We look for mitochondrial DNA copy number by qPCR. And for the DHODH clones and UMPS CRISPR clone, we have a decrease of DNA copy number. Also in non-stimulated RPU-SG3 CRISPR clone, but as soon as we are doing a stimulation, that we have an increase that is still understood. So in fact, I select some take-home message. So we identify some rare pathogenic variant located in exon of DHODH UMPS RPU-SG3. The DHODH variant affects the enhancer sequence. The variant increase inflammatory cytokine and chemokine production. DHODH and UMPS variant affect enzymatic activity. The RPU-SG3 variant affect the respiratory chain complexes. These variants are inducing oxidative stress, and there is a lack of ATP production, which is mainly glycolytic. But if you want to criticize this work, it has been done at AC16, and AC16 is a cell line. It's a fusion of cardiomyocyte with fibroblast. So we decide to start with Joao to work on IPS on the DHODH family. So we got some blood, we isolate IPS, and I said we, but he did. He differentiated this IPS to cardiomyocyte. And as you can see, when you are looking for DHODH activity on the IPS cardiomyocyte carrying the CT genotype, you have a lower enzyme activity. You have also a lower ATP production, and you have also a decrease of the mitochondria membrane potential. So in order to validate our approaches, we select the iPS cardiomyocyte carrying the variant, and we restore on these iPS the wild type phenotype. And we repeat the experiment. So when you are looking for ATP production on iPS cardiomyocyte, an extra one. Okay. When you have the wild type, you may have change in ATP production when you have a stimulation. When you have the clone carrying the iPS cardiomyocyte carrying the variant, you have a decrease of ATP production. But on the corrected one, we do not have change. So that's the first proof that we restore the wild type phenotype. And here we look for H2A2 level. As you can see, there is an increase on the CT iPS cardiomyocyte, but on the corrected one, there is no difference. So it means that here we have a strong oxidative stress, and we do not have it here again. Joel look for also for HME, and he has the same pattern. An increase of oxidative stress here on the clones that do not have the mutation anymore. You have a lack of oxidative stress. So what we can say about the iPS approaches? We have also what the same. I didn't change it. Okay. We were able to get the same result on iPS cardiomyocyte, and we were able to restore the phenotype, also in iPS cardiomyocyte. The second approach that we did some exome sequencing on unrelated individual. So when you are doing these approaches, it's a nightmare because you need to sequence an higher number of patients, and we sequence some asymptomatic and CCC patients, and we were able to identify new pathogenic variant. And I just want to mention MRSP 18B, that has been one of our additional candidates. So I'm just going to talk about some result about Drosophila. Is it a suitable in vivo model to validate our gene or to test some drug efficiency? So we have a live imaging system. So you have here the cardiac system of the Drosophila. It's not an organ. It's like a tube, and you have the cardiomyocyte, and the tube is also pumping like a regular heart. There is several reasons to test the Drosophila system. First, there is no immune response, so it helps you to conclude your result. It's a live imaging, so you can easily analyze the result, and you have no blood. It's hemolymph that has been transferred to all the system. And what we have in Marseille, so we were able to look for this Drosophila heart. We can do some imaging, we can process the imaging, and we can study multiple parameters. And most of the gene, approximately 75% of the gene, are conserved between the Drosophila and the human. So it's a good way. to analyze the candidate gene. Because for Drosophila, there is now some RNA for most of the genes that has been subcloned in the Drosophila genome, and these RNA are under the expression of a GAL4 inducer. So by this way, when you cross these two fly, this fly will have your RNA and the GAL4, and by this way you are inducing the expression of the RNAi that will be expressed only in the Drosophila system. And that's what we did for the NRSP18B RNAi. We stimulate these Drosophila and we look for the multiple parameter, and we have an increase of the diastole diameter, and we have also a increase of the systole diameter, and we have a decrease of the fractional shortening after only 7 days. And in this model, after 7 days, we have a complete necrosis of the Drosophila due to the RNAi that we are tested. So, as you can see, all the results cannot be done alone. I have to thank all the people here in São Paulo and in France. I just want to mention, of course, my brother, Edesio, George, João, Luis, and I just want to mention also Deia Kuramoto. She liked to stay in the shadow, but I know she worked hard for this meeting. So sometimes Deia, you deserve to go out of the shadow. Many thanks for what you did. And yes, all this work cannot be done without funders. So we were lucky since 2010 we got multiple funders. Thank you.
Okay, Christoph, thank you for your presentation. It was really nice as well. You had there to make a good team, both of you. So now we are open for questions.
Very nice talk. Very interesting. I have a question about you. Probably I didn't understand well. So when the individuals carry the mutations that you describe, they're fine as soon as they develop Chagas, right? But then in Drosophila, if you do have the mutation per se, you already have a phenotype, right? So what is the difference that you...
Okay. When we did the study, sorry. When we did the study on the family, we identified some patient carrying the mutation, but the Drosophila do not have the mutation. We just inactivated the candidate gene. It's really difficult to do CRISPR to introduce the mutation in Drosophila. There is a limited number of papers on CRISPR on Drosophila. All the papers come from one group, and I know that multiple groups over the world try to do CRISPR on Drosophila and it never succeeds. So I agree, it's not when we are doing RNAi on Drosophila, we are not looking for the variant, we are just looking for inactivation of our target gene.
I have a question first, sorry. Christoph, how can you translate the evaluation of the heartbeat in the Drosophila in comparing to the human in your strategy to test drugs and translate the results from one model to the other?
What can I say? I have some limitation. We have identified some drugs, and when we test these drugs on AC16, it's clearly we clearly demonstrate that we have an improvement of the ATP production. Some of these drugs has been tested in Drosophila with a high sugar diet, so we induce a cardiomyopathy in this Drosophila, and the same drug restore the wild type phenotype. And on these Drosophila, we have parameters that we are looking for that are completely changed, and the drugs restore the original value for these parameters, and it's consistent. We have some parameters linked to arrhythmia, and we have some parameters linked to fibrosis, that we need to push forward.
Do you have any idea of how nitrooxidative stress is damaging mitochondria? If it's just on my lipid membrane, if there is a damage to DNA, because you said you have decreased mitochondrial count.
Yeah.
The assembly is compromised.
Good question. Yeah, as soon as you have an oxidative stress, your mitochondria are abnormal. You do not have ATP production anymore. You do not have the gradient of electron. It's a big mess. You have a lower copy of mitochondria. Probably the fusion and the fission process are completely modified. So it's another pattern, and it's difficult to recover from this.
Yeah, if I can just complement.
Yeah, sure.
So Luis has done some experiments looking for oxidized mitochondrial DNA, and it kind of parallel. So we have a decrease in total mitochondrial DNA in the iPS cells with the variants, but you have also an increase in oxidized mitochondrial DNA. So, but then it's not necessarily the mechanism or the only mechanism for mitochondrial damage, but that's something that we're looking for.
We still have some minutes for more questions.
Correct me if I'm wrong. If you have like a mitochondrial dysfunction, in other words, a more glycolytic phenotype, you are more susceptible. Thinking about the cardiomyocytes, they are more susceptible for interferon gamma deleterious effect and also to Chagas disease, right? Or
well, you have a main glycolytic sources of ATP, but whatever your respiratory chain is completely destroyed. So by this way you are producing ATP, but you do not have also fatty acid oxidation. So what I will say, you produce ATP glycolytic ATP, but it's not enough. Who cares? Because all the system is blocked.
yeah, what I'm wondering is like thinking about a normal person without any mutation that leads to mitochondrial dysfunction. Like for example, if you treat cardiomyocytes for a normal person with dichloroacetate, for example, to push oxidative metabolism, can you like induce any cardioprotection by just pushing oxidative metabolism? Have you guys like tried to do that or thinking about doing that? Thank you.
Good question. In my mind, at the beginning, I was thinking if the respiratory chain is not working correctly, maybe the fatty acid oxidation will go up to produce more ATP that will balance the phenotype. But in fact, the proteomic data shown that fatty acid oxidation process is dead also in the CCC. You want to add something?
So yeah, in heart failure in general, you have a shift from away from fatty acid towards glycolytic metabolism, right? And we show that with cytokine treatment of cells, we have a similar picture. And yes, we really want, although the current metabolic therapy for heart failure is actually based on increasing the glycolytic metabolism and suppressing more the fatty acid oxidation. And this has been done because this way you will get ATP with less oxygen consumption. And this is a big problem for ischemic disease. But in Chagas, it may be, in other inflammatory situations, it may be the opposite because there's not a big issue about loss or reduced oxygen release there, but we have this decreased fatty acid beta oxidation. So we are considering using drugs that can increase the fatty acid oxidation per se and oxidative phosphorylation as well.
And we have this increase of glycolytic ATP, but we must keep in mind that the total ATP production is down anyway.
Just a quick question. Have you identified any homozygous individuals for those mutations? And do they have worse phenotype?
Okay. So we have identified 22 variants, and in each family, the individual were heterozygote. For the DH variant, this DH variant was also associated to the Miller syndrome. And in the Miller, it's a neurodegenerative syndrome. And the children affected are homozygote for this variant, and they do not survive for a long time. Yeah.
So this Miller syndrome patients, apart from the facial cranial abnormalities, 70% of them have congenital heart defects. So the variant is important in heart morphogenesis. And the other thing, just a quick one, Christophe showed that the family tree. So we had a family with a symptomatic mother without the variant, all the cardiomyopathy patients with the variant, and 2 seronegative children who were probably born after the family moved away from the endemic area. And one of them had carried the variant, and the other didn't. And we had when we did the iPSC experiment, we had the same phenotype. So when we added the cytokines to this iPSC from this CT carrying normal sibling, there was a reduction of ATP production. But along his life, he was like a 35 type. He had no history of disease. He was eutrophic. He had no history of disease that could be associated with the mitochondrial phenotype. Right. So it's that's what summed up to our hypothesis that you only get the mitochondrial phenotype in the heart disease if you have inflammation on top of the variant.
And to complement, just 2 sentences. For the DH variant, as you see, it's probably affecting an enhancer sequence. So we have now this variant subclone in cardiomyocyte in the X616, and we are going to get soon this variant introduced in neuron. So by this way, we will be able to look for the enhancer activity in cardiomyocyte and in neuron. And we will look for the target gene of this enhancer. And probably it will help to decipher the pathogenic associated to the cardiomyocyte and to the Miller syndrome.
Okay, so we are out of time. Thanks, Christophe. And now we are going to the coffee break, the second one for the day. And we see in the next 30 minutes. So let's continue with the workshop. And now let's hear from Professor Daniel. He's a biochemist trained at the University of Campinas, where he earned his PhD. He completed postdoctoral research at the Max Planck Institute in Germany and the University of Cambridge, and later was a principal investigator at LMU Munich. Since 2014, he has been a professor at Unicamp. His work focuses on neuroproteomics and the biology of psychiatric disorders. Oh, please start your presentation and welcome.
Thank you very much. So good afternoon, everyone. Thank you for being here and for the invitation. Adesio, Sovan, for organizing this. Very top-notch speakers. I'm really glad to be among them. Thank you very much. So the idea here is to tell a bit of a story of what we are doing there at Unicamp in terms of using this iPSC-derived cells to understand schizophrenia more specifically. So, but just to start, why studying schizophrenia? It's actually a disease that affects a lot of people in the world. Actually, 25 million people around the world has schizophrenia, and it's an incurable and chronic disorder, meaning that it's uncurable. They have to take medications to deal with the symptoms. But actually, symptoms-wise, there are so many symptoms that actually they're divided in different classes of symptoms, which will progress during the course of the disease. Normally, it starts with hallucinations, delusions, and this will progress then to cognitive symptoms and the so-called negative symptoms that will then lead to a very poor type of interaction of the person with the world. These people, they normally cannot study or work properly, and schizophrenia is among these psychiatric disorders which are the most disabling of humankind. Actually, a person with schizophrenia loses about 30 years of their lives. Apart from the fact that these people live less, they actually cannot work properly, relate with their relatives. They are fully dependent on either hospital services or their own family. This is a burden for the family as well. And meaning that we have to study more about the biology of this disease to better understand it and, of course, bring up better treatment strategies. Actually, about half of the people that are treated for schizophrenia do not respond properly to the antipsychotic medication, which are normally used, meaning that we do have to do something in terms of understanding this disease from the molecular and biology point of view. And as I said, as part of a story that I'd like to tell you today, we've started working with schizophrenia brains actually in 2004. It's about 20 years ago. During my PhD, I started looking at proteins which might be differentially expressed into tissue collected post-mortem from people with schizophrenia and controls, and eventually other psychiatric disorders as well. And for that, we used proteomic approaches. And I think everyone here appreciates, we've already talked about proteomics here today, how important this would be. Actually, the dynamicity, how dynamic actually proteins are in terms of tissue and eventually in front of a disease or treatment. This fluctuation of proteome might be interesting in terms of information that you can take out of the biology you're studying there. So, and then, as I said, we've looked to the proteome of several brain regions from people with schizophrenia. We mapped these proteins there, and we found a bunch of differences around these different brain regions we've studied. So, for example, proteins associated to energy metabolism, synaptic deficits, cultural circuitry, and also glial dysfunction, which was at that point something new in the field because we were very much, still are actually in schizophrenia research, very neurocentric. But glia cells have actually a very important role in this disease and also in other psychiatric disorders. And we are interested in looking a bit on that. Interesting to say that actually all these facts were not unknown to schizophrenia. But the thing is that with proteomics, we could point to some molecular targets or proteins which are involved specifically to that. We are studying it, or even pathways which are can be targeted for broader or deeper investigations. But of course, as we are looking in tissue from people, now if we want to validate the importance of those proteins and pathways, we have to model the disease. And we cannot do that in living people. Of course, collecting brain tissue from living people is not an option. We cannot treat people and test a protein, for example, in someone who is living, and then we have to model this disease. As we know, most of the preclinical model work will be done then in rodents, and this would be like the gold standard for investigating, for instance, a brain disorder. But for schizophrenia and for psychiatric disorders in general, it's a little bit hard to model a disease because we do not have something that is visually possible to say, here I have a schizophrenic mouse, for example, right? Because we do not have any modifications in morphological terms. And actually, even the behavioral tests that we can do, that one can do to characterize an animal model, it's normally not applicable or not specific for a given psychiatric disorder, meaning that eventually you could see a behavior in the mouse which could overlap to a model for depression or for schizophrenia or for bipolar disorder, meaning that it's a little bit hard to go with that. Of course, there's people studying that and doing work on that, but we were a bit restless on only looking at that. We did investigations on animal models. I'm gonna even show some data here with that, but we thought about moving on to in vitro preclinical models and eventually to models that could be a little bit more sophisticated than a normal cell culture, for instance, or cell lineage, and that could make use of the genetics of a patient, okay? That's when we started working with then iPSC-derived cells or induced pluripotent stem cells. We had a collaboration that actually started with this postdoc called Juliana Nascimento. Juliana started in my lab here when I started my lab in Brazil 10 years ago. And she bridged a collaboration with Stevens Rey in Rio de Janeiro at the Federal University of Rio de Janeiro, where they actually had access to iPSCs derived from patients with schizophrenia and control, people that are neurotypic. Meaning that they had these iPSCs in the lab, and from these iPSCs, we could derive brain organoids and other neural cells. Of course, brain organoids have their limitations. It's a nice model, you know, it's something somehow impressive if you have like a kind of a mini brain in the lab, but of course there's a lot of caveats as well. I mean, they're not completely matured cells, not vascularized, the type of organoid we do. Nowadays, there are some people doing out there organoids that are vascularized, but we don't do that so far. We do not have all cell types anyway, but it's at least an option which we can work with a cell from a patient and make that something for building up biology around schizophrenia in that case. So this is how they look like small spheres, and in terms of the morphology, they look like brain somehow. Stevens' lab that we were collaborating with, they had done several validations in terms of the morphology. And the first question Juliana had was like, do these cells or do these organoids look like brain in terms of the proteome? As we were investigating proteomes since a while, brain proteome, we were comparing then now the proteome. The first thing we would do was generating organoids out of iPSCs, independently of being from schizophrenia cells or not, but just looking whether these look like a brain proteome. And yes, actually the answer is they do. We could see several biological processes and markers which are generally present on neural cells. For example, all the classical neuronal markers, also progenitor cells, neural cells, and astrocytes, and also markers for oligodendrocytes. These were actually 45 days organoids, which you could even see some markers from oligodendrocytes, which are not generally easy to get, but Juliana could establish that well there. Not only that, but just to complement here, actually, we could identify among the proteome of the cerebral organoids in the lab the complete set of proteins which are part of glutamatergic transmission, dopaminergic transmission, meaning that we would have like an interesting model there to study schizophrenia. Then we moved on to then do schizophrenia patients derived organoids, neurons as well, coming out of those iPSCs. And of course, the first thing was doing a proteomics to compare that to non-affected cells or control cells. And the first idea we had here was that when we were doing the proteomics of brain tissue, every time we were presenting this data or going to publish that, what we've heard also always was like, how do you know whether these, how can you guarantee that these proteins you were seeing differentially expressed in those brain tissue are pertinent to schizophrenia and not, for example, anything related to the postmortem time? Because when I say postmortem brains, this means, in our case, brains that were collected after 15, 20 hours, after 15, 20 hours after death, right? Then how could we discard, whether those differences we were observing were associated to schizophrenia or actually to something associated to the postmortem time, right? And then this was something that we wanted to answer with these organoids, whether like if we do a differential proteomic analysis here, can we see the same or at least something similar to what we saw in the postmortem brains? And that's why Juliana did these organoids there from schizophrenia, and we've characterized them with the main markers to see that they were developing properly 45 days after the protocol of the 45 days. And long story short, yes, I mean, these differences observed in the proteome of schizophrenia organoids compared to controls were similar to what we saw in the postmortem brains some years before, right? And then there were of course, some differences which were specific to each type of cell, but the main differences that I showed you before associated to glia cells, energy metabolism, they were all present in the organoids as well. So then letting us somehow more comfortable in saying that this is an interesting model to study schizophrenia and eventually treating with some stuff or some medication, possible chemicals for understanding better new medications or even the role of the known medications which had been used since decades, but eventually we don't even know what they do specifically. So, but then we started digging a little bit deeper on the data we had previously, trusting now that these are some roads that we can travel more safely. And then energy metabolism-wise, we wanted to take a look at some of those differences. And though for whoever works with proteomics, these energy metabolism differences, they are pretty common, because whatever you're going to study in terms of proteome, energy metabolism proteins, they are very abundant and they are well ionized somehow. They are all the time appearing. If you get a person working with bacteria or plants, whenever energy metabolism is involved, we are going to identify them. But for us, there was an interest about energy metabolism because there is a whole theory about schizophrenia associated to the metabolism of the brain. And then in the 1970s, there's this paper that comes out and actually comes up with this theory of the hypofrontality, meaning that a person with schizophrenia had a lower flux of energy in the frontal cortex, which is the part of the brain that makes us humans, actually, right? Then for us, it was important to dig a bit more about this energy metabolism part. So then Juliana was doing her master's there in the lab, and she generated neurons derived from iPSCs, from schizophrenia cells and controls. And the idea was to look a little bit deeper on the mitochondrial proteome. Actually, Juliana enriched nuclei and mitochondria from those neurons that she generated from iPSCs, from schizophrenia cells and controls. And she's done a proteomic analysis, a subproteome analysis from mitochondria and nuclei. And she's done some other validations afterwards. But one interesting thing that she found is that, of course, she looked also at the whole proteome of those cells. She found some differences found proteomic differences when comparing neurons from schizophrenia cells and controls. And she saw that actually the differences on the nuclei proteins eventually some proteins from the mitochondria are very much connected to cytoplasmic differences associated to axonal guidance. And we do know from the literature and from our data that when these neurons are developing, they do not branch as a control neuron. Actually, they have less connections, let's say. And I'm going to show you that actually these neurons, schizophrenia neurons, they actually spend more energy and they branch less. They communicate less, meaning that they struggle somehow to minimally develop. And one of the things that might be involved with this developing is axonal guidance. And we have a whole set of proteins here that connect proteins that are controlling this from the nuclei and signaling this from mitochondria to the cytoplasm, showing targets why these cells might not be developing properly. Another fact that we thought interesting to see was in the course of differentiation, actually, these are neurons derived from iPSCs, but of course the first step is to make these iPSCs neural stem cells and these neural stem cells, we drive them for neurons to become neurons. And in the course of differentiation to a neuron, we see that this glycolytic enzymes, there's like this shift on glycolytic rates. It's known from the literature that actually glia cells contribute a lot to the energy supply for neurons whenever they become specialized cells. And this includes this shift to less glycolytic rates in terms of developed or mature neurons when compared to neural stem cells. And while we see that in control cells, we do not see the shift happening in neurons from schizophrenia cells, meaning that this could be the reason why we see some of the things that are known already from the literature about schizophrenia and about having a higher energy environment, which might lead to oxidative stress and DNA damage, because these cells should shift for somehow producing less, having less, lower glycolytic rates, but they still do the same rate as they were before differentiation, and this could somehow exhaust the whole system. And Juliana did some analysis on those differentiating neurons. As you can see here, respiratory-wise, these cells, they somehow struggle to develop. As I said, they actually spend more energy, much more energy, but they do not properly develop as like a control cell. And in terms of redox state, apparently this is a very simple type of analysis, but apparently these schizophrenia cells also have some higher redox states, or at least what we could infer being something like an oxidative stress. So, but that's this one way we went. Then the other thing, as I showed you, that we found in the brain tissue there and also observed in the organoids were proteins differentially expressed associated to glial dysfunction. And these we could focus specifically from what we see in the organoids in astrocytes and oligodendrocytes. As you guys must know, we have like two types of cells in the brain. There's glia cells, neurons, right? And glia actually is the Greek for glue. And then these were cells that 20 years ago, people thought they were much more like tissue that only holds somehow the neurons. But I would say from the last 2 decades to now, more and more we're realizing how important these proteins are in terms of brain communication and neuronal support. Our data and others actually have supported the idea that schizophrenia might be actually a disease from glia, and then actually the fact that the neurons connect worsely in schizophrenia might be a reflex, actually a consequence of the fact that glia cells do not work properly there. And that's why we want to investigate it a bit more of these proteomic differences we've seen there. And as I said, the telling a story since the beginning when we started with the brain tissue and moved on to preclinical models, our idea was when we did a whole proteomic analysis on postmortem brains, we were looking at some different brain regions in a lot of proteins, doing some shotgun proteomics. And the idea was trying to then move towards validating the importance of pathways and importance of specific proteins regarding schizophrenia, meaning that we have something more targeted, looking at less proteins, but understanding better what they do, actually, or their importance in the disease. And then we've started looking, as I said, firstly, as I showed you with Juliana work was something more related to energy metabolism in general, but now we wanted to look more into proteins which might be specifically associated to schizophrenia. And as I said, in the context of glia cells, oligodendrocytes are those cells that actually insulate the central nervous system, the CNS, and doing like myelin, like putting myelin there. Actually, it's like an insulation, really. It's as you see, like a wire in electric wire, which we insulate, not for not being getting a shock there, but the thing we put that rubber around it just so we don't lose energy that is passing there. And it's the same in neuronal transmission, because we have like transmission of electrons there in the axons, and we have to insulate them with myelin so they can be properly transported there. And it's known that, as I said, several molecular findings from proteomics, transcriptomics, and also some other findings in terms of the morphology of brain in schizophrenia has shown that a person with schizophrenia start losing myelin not like as in the same rate as a demyelination type of disease, but they lose myelin. They have less myelin than a normal or a neurotypic person. Then this shows the importance of these cells in this context. And Carol actually was, during her PhD, start working with a family of proteins, and specifically with one of protein of these families, HNRNPs. And these proteins have a lot of distinct type of roles in a cell, things from controlling splicing and also signaling roles in the cell. But more recently, it has been found that these proteins in the brain, they also control somehow, or they have some control towards myelination. And Carol was more interested with this cell, with this protein, because we found this family of proteins differentially expressed in pretty much every type of proteomic analysis we've done. Since brain tissue organoids or other cells that we've differentiated in the lab, we always saw these proteins differentially expressed. And she wanted to understand a bit more about what this protein is doing there, specifically considering that it controls myelination in the brain. So what Carol did then, she worked with a model, a Cuprizone model, which is a demyelination model. It's a very classical one. People working with multiple sclerosis, for example, are using this type of model, which consists on feeding a rat or mouse with this with a food that has cuprizone on it. It's a copper chelate cuprizone, and this leads to demyelination. But the thing is that you can feed this animal for 5 weeks with that, and after 5 weeks you see a broad demyelination. It's something like that. You can see like in control before this 5 weeks of feeding with cuprizone. 5 weeks after, you see demyelination very clearly happening here. But then the interesting thing is that if you stop the food and start feeding with the normal food, 5 weeks after, it reamyelinates. You know, it comes back to not normal, I would say, but reamyelinates. And it's something that we can see here. It's like you can see reamyelination is a little bit more, it's a little bit stronger somehow. Of course, not as it was previously, but there is some remyelination. And the question that Carol did was like, if that protein is associated to demyelination or to myelination somehow, what if we, after in the course of remyelination, we somehow block the protein? In this case here, she started thinking about using some genetic modified model, but she ended up using an inhibitor of this protein there in the course of remyelination. Can we see something that might be similar to what we observed in schizophrenia regarding myelination? Because again, we found proteins associated to myelination in the postmortem brains, in the organoids, and also we've already seen and have done some work in vitro showing that several of the most the best treatments with antipsychotics promote myelination. And then we wanted to see how this protein could be imported there in this scenario. So, and then what Carol did was like she fed these animals with cuprizone for 5 weeks, and then she performed some behavioral tests with these animals after 5 weeks. And then she used this inhibitor for this HNRNP-A1. It's one protein of this family. And in the course of remyelination to see after that whether this could impair really remyelination as you would expect. And then she's done proteomic analysis at the end and behavioral tests again to see what happened. So for not our surprise, but somehow our surprise, we did not see many differences in terms of the behavior after she used this inhibitor during the course of remyelination. And it was something that at the beginning, this was the first data we had, and then we thought like maybe the protein is not that important, right? In the context that she was asking about. But when we looked at the proteome, there we see a lot of differences, you know, even not observing behavioral differences in molecular terms, we see a lot of proteomic differences after the remyelination with the inhibitor of this protein there. As said here, we had like 3 different regions being analyzed, about 350 proteins differentially expressed, and several of them associated directly to myelination status, meaning like the classical markers of myelination, and all of these proteins somehow associated to myelination. Okay, this is a circus plot associated to myelination proteins. And this was something interesting for us to see and interpret, because the fact is that then we've done some validations afterwards just to be
confirming these differences there. But there is a behavioral and proteomic discrepancies because we can see the molecular differences and not behavioral differences. But it's important to say that schizophrenia is a disease of the neurodevelopment, meaning that normally young adults are affected by schizophrenia, meaning that they get to the hospital and be diagnosed with schizophrenia. Normally people about 16 to 25 years old. And when they go to the hospital, they have severe they show severe symptoms already, and they are very strong. I mean, there's no like symptoms start appear and then suddenly there's like a hallucination or something like that. These people, they are somehow normal, and suddenly they have like a psychotic breakout, you know. And then when they are diagnosed, it's what is thought about to be is actually these people are sick since a while, you know, but they do not exhibit symptoms, meaning that they are probably this theory of neurodevelopment is that probably something happened during the neurodevelopment in utero, right? During the development of the brain still in utero. But then they have this chronic disease going on, and then at some point the symptoms break, right? But and this neurodevelopment theory is also accompanied by the fact that it's a multifactorial disorder, meaning that if you look at genomic loci implicated in biology of schizophrenia, you see in this study here, they looked at 300,000 people, and they found like 200, almost 300 genome loci, and about 120 genes that might be involved. And meaning that you don't have like a gene or a couple of genes involved in schizophrenia. You have a lot of genes involved with that. And what is, and this, you know, this is being published in 2022. And since 20 years, I would say, people are looking for genetic markers for schizophrenia, and they did not find it, you know. They could only find when you see something like more of a technology developments allowed then for you to look in a lot of people, a lot of genes, meaning that we might have something here that since the neurodevelopment, you have a lot of genes being acting in a small effect, you know. And then the thing is that after a point, you see that all these differences that these genes might carry over the years together will break out in a kind of a psychotic symptom. And we wonder then whether this protein wouldn't be one of those important targets, right? Because we see that when we mess with it in terms of myelination, it does lead to differences on molecular terms, but not in the behavioral terms, meaning that somehow it's not strong enough, let's say, to change the behavior of the animal, but somehow it does change something molecularly, right? Then we thought about that this could be an important protein that among several of them in the course of the long disease, a chronic disease, could be an important in the context of neurodevelopment. And some here, but some other stuff we're doing also in the astrocytes, looking at proteins associated to astrocytes, also astrocytes derived from iPSCs, from schizophrenia patients and controls. Veronica did her PhD looking at that. She was more interested on this protein called, this enzyme called phosphoglycerate dehydrogenase. Again, also a protein that we found differentially expressed in several different type of cells and tissue we've analyzed. And long story short, what you could see is that this protein is quite important in the production of serine. And when she's generated neurospheres from these proteins, and she looked from different angles, from the metabolomics angle, cell death wise and everything, we could see that actually inhibiting this enzyme in a neurosphere, for example, lead to something like that disturb the neurodevelopment, because we have like a neurodevelopment type of model here when we do neurospheres, because we start from a neural stem cell and get to a neurosphere. And then we are somehow simulating the neurodevelopment. And we see that serine metabolism is very important on this regard, which is not a novelty, but at least we are pointing specifically that this enzyme, which we found differentially expressed in different tissue we've analyzed before, seem to be pivotal for that. But summing up, as I said, we've done a lot of brain proteomics in schizophrenia, employed some preclinical models, including animal models, but mostly in vitro models, as I showed you here, from the proteome to pathways, looking at the energy metabolism. Of course, it's something important. And again, the importance is that we know since 50 years that energy metabolism is important in schizophrenia, but looking at proteomic terms, we can look at molecular targets for that. And as I said in the story of HNRNPs in oligodendrocytes, how these proteins can be one of those important markers in the course of neurodevelopment and could be somehow silently being associated to myelination. And I couldn't tell you much about the phosphoglycerate dehydrogenase in astrocytes, but as I said, it's an enzyme very important for the production of serine, which is important for glutamatergic transmission. And we could see that this is important in the context of schizophrenia. There are some other chapters in the story. I hope I have time the next time to tell you about it. We look for schizophrenia treatment biomarkers. Basically, we're looking to see whether we could predict with lipidomic and proteomic signatures from the blood whether someone is likely or not to respond to schizophrenia. This is something that Leicia has been doing during her PhD. She has a kind of a multi-omic type of analysis on lipidomics and proteomics to try to come up with biomarker panels for that. We have some people working on antipsychotics and CNS adipocytes. The crosstalk about these two, actually Julia and Brad are playing with treating adipocytes and neural cells, and eventually use the conditioned media to treat one another and see how the treatment with antipsychotics could also lead to the metabolic syndrome that actually antipsychotics from the second generations generally do. And we're interested also in studying endocannabinoid system. It's something that was brought to my lab about 6 years ago from a postdoc, and there's still a lot of people interested in that. We have some work on oligodendrocytes and neurodevelopment, how endocannabinoid system might be one important aspect of that, specifically saying because now we have in clinical trials cannabidiol for treating schizophrenia, right? And we have Dani here that will be collaborating with Solvon, hopefully. They're going to his lab, working on energy metabolism, a crosstalk about energy metabolism, autophagy, and how CBD can be involved in that, or can modulate that in astrocytes, derived or not from schizophrenia people. And more recently, we're looking also neuroinflammation-wise in microglia. Aline is doing that during her PhD, and meaning like we're moving on and understanding a bit better and a bit deeper data that we've generated from proteomics since a while, but now we want to investigate it more, focusing more on those proteins we found since some years ago. And just to do some final advertise here, actually, Guilherme was a PhD student in my lab. He finished last year, and we published a new way of analyzing proteomics data. It's like you can find here, like in a web-based type of software, which we can just throw your proteomics data in there and get a lot of information from it. I think it's an interesting tool because it congregates a lot of different algorithms to analyze proteomics data. And more than that, it's very user-friendly. You know, it's not something like static that you cannot move things around. You know, you can pretty much edit your own figures. It has like a module where you can do whichever type of proteomic analysis you want to do it. It's very nice. If you are interested to do it, I invite you to take a look at this tool that he developed. And just to finish, thanking my collaborators. As you guys know, we do not do science without collaboration from everywhere here in Brazil and abroad. And Solvon specifically that is here, as I said, maybe next time I can show Dani's, or Dani can show her data here that we are going to work with together, and my group in Campinas. And thank you very much for listening to me.
Okay, thank you, Daniel. Questions?
Thanks, Daniel, for the wonderful talk. I have a couple of very basic questions. So first is, from all the, proteomics analysis and the, studies you have been doing over the years on schizophrenia, have you come up with a drug target, or apart from cannabidiol, what do you think could be used for treatment, or is there something in development? And the second part is, do you think, apart from proteomics, can you integrate multi-omics data to have deeper insights?
Thanks, Owen. Actually, I wouldn't dare saying that we found some targets for treating schizophrenia. I mean, we've done some very basic in silico analysis with this, all this proteomic data we have. We've already reviewed that in rerunning our previously obtained data and more recent obtaining data with, as I said, some in silico platforms. We did find some potential drug targets there, but we haven't tested. It's I think we're it's something that we could explore, but we didn't. As I said, I think the closest that we get from treatment was, as I said, trying to look for these signatures in blood, lipidomic and proteomic signatures in blood. Though one may say, like, well, you're going to get signatures for treatment that actually do not work properly. But that's the thing. These are the only treatment that exists, right? And these are the treatment that people still fail in having it. Meaning that we wanted to see whether we could contribute to giving an answer that could be useful for the clinicians, to say, like, at least saying, this person had a lipidomic signature in the blood that says that this medication won't work. Then you can go to another one, right? Because the thing is that a person that takes an antipsychotic medication, it takes at least for 6 weeks to see if they improve. And if they don't improve, they have to change medications, and again, another 6 weeks. And in the course of these 6 weeks, they start having these psychotic breakouts, and this will lead to cognitive dysfunction that is not recoverable. And then it's very imperative that they find the right medication since the beginning, if they want to be, like, properly medicated. And as I said, half of the people do not find that, meaning that half of the people will have severe cognitive dysfunction at the end. And then this is something that I would say we contributed somehow, because some of the data we've obtained that we had a patent. We have a patent for that, for this signature. And we had funding for developing a product, from FAPESP has a funding about small companies, like startup companies. And we were trying to then move on to have, like, a test whether it could be useful in the clinic. And cannabidiol is something that we have a very strong group of people in Ribeirão Preto doing that. We collaborate with them, and they are doing these clinical trials on CBD, on cannabidiol. And these we are studying from the molecular point of view, giving, like, more substrate, specifically in terms of the effect over glia cells. I mean, these are our contributions, but mostly not looking specifically to drug targets that could be useful or repurposed in terms of chemicals that are known before. Oh, you asked about you also asked about the multiomic thing. Yeah, Licia is the student of mine. She's working on that in her thesis, and she's trying to integrate now lipidomic and proteomic data. I have another student who is Vitor, and Vitor has, like, a more difficult task, which would be proteomic, metabolomic, and also single cell sequencing analyses, to try to integrate. He's just started his PhD, meaning he doesn't know how to do that, but he wanted, I convinced him that he had to do it, and he liked the idea, And I did say, if you do it, you're going to be, like, one of the first to do it. He believed, and then let's see what he can do. But yeah, well, he likes it, but he knows how to program, he knows he likes dealing with the data, with large scale data, and he's convinced he can do it. Let's see what's going to come up. But we still don't have any solid results about it. Yeah.
Great talk, man, as always.
Yeah,
I have a question which is linked to the previous question. So you showed that iPSC-derived neuronal cells from schizophrenia patients, they don't have the metabolic shift from glycolysis to, but they already have increased oxidative phosphorylation, whatever.
Mm-hmm.
So there is a clear, not damaging, but disruption of this shift.
Uh-huh.
And then you showed that in astrocytes, they have impaired also in the serine metabolism, which also comes from glycolysis.
Mm-hmm.
So is there any way that you can measure how is this flexibility in metabolism? So how the cells can travel from glycolysis to oxidative phosphorylation and then provide all of these elements for keeping the system working using all this omics approach that you're...
I don't know. It would be great,
'Cause this
actually.
is-
Yeah. Actually, it's as you said, there's like a clear connection of those two entities, but I don't help me how to interpret that. It was something we can try to talk about later. I don't know. It's definitely something there, but I wouldn't say how to. I wouldn't say how can we do that, you know, or how can we explain that, you know. What could have started first, right? Is either if this has like the origin in astrocytes, and this will somehow, I don't know, make neurons need to do more energy because they do not get enough from astrocytes. I don't know.
Yeah, and because they need to change the metabolism according to the demand. So of course, if ATP is higher, so but if you might produce some amino acids that you need to shift the glycolysis to. So this is something that we can talk about.
Yeah, sure. It would be great. Thanks.
Awesome.
Yeah.
Okay, we are out of time, and we appreciate Daniel's presentation. Thank you, Daniel. Now I would like to invite our last speaker, is Professor José Eduardo Krieger. He is graduated in medicine at Ribeirão Preto Medical School of the University of São Paulo. He earned his PhD in physiology at the Medical College of Wisconsin and completed his postdoctoral training at Harvard and Stanford. Also, he is a member of the Brazilian Academy of Sciences and the São Paulo Academy of Sciences, and leads the Laboratory of Genetics and Molecular Cardiology at INCOR. His research focuses on molecular markers in cardiovascular diseases and innovative therapies for cardiac regeneration. Please welcome Professor Krieger.
Hi, good afternoon. I first want to thank Edesio and the colleagues from Birmingham for the invitation. It's pretty nice to be here. So we're going to shift gears a little bit, but the challenges are the same. So you're going to feel familiar with the problems that we face. So the idea is use cell model systems to try to understand better what's going on with several cardiovascular diseases. For the sake of this discussion, I will focus on post-myocardial repair. So we all know that cardiovascular disease is quite prevalent, and if you have an MI, you get some type of adaptations, but it's very likely that if it's a big MI, since you cannot replace the cardiomyocytes, that you're going to end up with heart failure, which is a disease that has very high morbidity and mortality. So it's a major issue for the individual, for the health system, and for society as a whole. So nowadays, when you have this problem, we have an array of interventions that can be done: surgical, pharmacological, hemodynamic, and so on. But still, and we have several ways of assessing initially or late if things are going well or not. And for instance, if you have a chronic heart disease, and you can assess an index of ischemia, global ischemia. So you do an MRI, magnetic resonance imaging, you assess 17 segments of the heart, and if you look at them all and we summarize, we say that we have a global assessment. So when you are a candidate for a bypass surgery, you have a high index, ischemia index, and as we all know from the 70s, you get the graft and you improve, and this improvement, it remains for years to come. But you see that you don't normalize the problem. And lately we have been trying different approaches to improve this situation. I mean, either when you were talking about hemodynamic placing of a stent, or even a combination with the cardiovascular revascularization, and we can get this type of data. For instance, injecting mesenchymal stem cells, in this case, bone marrow cells. So while this is the global ischemic index, here is the segment one, where you inject the cells. So, and we and others start to get evidence that you could, in fact, increase tissue perfusion. So we're not talking about improvement of heart failure or anything else. We're trying to answer a simple question. If you inject the cells, do I have or not an improvement in tissue perfusion? For that, you can use few of these techniques. One of them is magnetic resonance imaging. And we went on to even establish a physiological parameter, which is coronary flow reserve. It's more or less your capacity to dilate when you need the heart to do that. So if you have a problem, your heart, the vessels will not dilate. So this is not structural measurement. This is functional estimate that things are improving. And again, we detected that injecting the cells in the regions that receive the cells, you could do that better and sustain for even up to 12 months, which is quite impressive. In the adjacent area, you saw similar results, but at the distance not. So indicating that some of the things that we still don't know how they work, I mean, they will not take someone from the line of heart transplantation. That's what we expected early on, 15, 20 years ago. But we are showing that you can indeed alter the microenvironment and eventually have an improvement such as the ones that I showed. But essentially, the challenges are the two aspects. One is replacing muscle. We are not doing that yet. And the second is trying to improve the reparative response. And here, eventually, we can identify two big targets. Endothelial dysfunction, because in addition to a major block of an artery, you may have a disease in the microcirculation, and that's a problem. So if I go there and I do imaging, I don't see any problem, but you don't have the performance. A typical example is someone that has refractory angina. I mean, it's a person that has a terrible quality of life. You go there, you can image, there's no vessels blocked, but the person, it's very limited. And the second is fibrosis. And fibrosis, remember, is an essential component, because if I don't have fibrosis, I will have a hole, and that's not very good. But if I have too much fibrosis, or these complex patterns that you can observe, with time, they are very bad. And we are trying to then use these model systems to understand these processes. I will focus on this discussion, the endothelial dysfunction and the scar remodeling. And of course, the regeneration is a different chapter, and we have not progressed too much on that arena. We could recapitulate these observations in the pig. The pig for in the cardiovascular is a very good surrogate of human beings, not only because of the size, but because of the function of the organ. And as you can see here in this protocol, it's a 4-week protocol, and we test different doses of cells, in this case, mesenchymal cells from fat, from a single donor. We injected male cells into females because we want to see if we could find the cells there. 4 weeks later, I'm going to tell you, we did not find any of the cells. It's not that we are looking for imaging cells. I mean, we assayed for Y markers and we could not find. But the interesting part is that, again, we observed the highest dose an increase in tissue perfusion. In this case, was not MRI, was echo with microbubbles. So those are two techniques that allows you to estimate perfusion, which very difficult to perform in rodents, rats or mice. We use surrogates. We look at if VEGF is increased, markers of endothelial cells, and we assume that the blood that the flow has improved. But this is more a direct assessment. That's very important. In addition, in this case, we demonstrated that at the highest dose, we decreased the size. This is a small MI, about 11%, 12%. So we have an addition decrease in 5 to 6%, which is very significant in this case. But most important, in addition to this large change, if you will, in the fibrosis, we also observed that you have micro changes, if you will, meaning that the quality of the fibrosis was altered in the animals that received the cells, meaning we shift the balance between mature and immature fibers. And it may be very relevant because the physical characteristics of the immature fibers are very different from the mature. While the mature are strong to fill the hole, if you will, I mean, they are mechanically, they are not very friendly for an organ that has to pump 60 to 80 times per minute, hours, days, decades, and so on. So this may be something that's quite interesting that we had not observed before. So then fibrosis is a big issue, is an independent predictor of the adverse outcomes and mortality. And there are several ways by which fibrosis can impact the cardiac function and result in the adverse outcomes. So in order to try to understand the underlying molecular mechanisms associated with these beneficial effects of mesenchymal cell transplantation, so direct inject into the muscle, we start to use this model system. So we and others using or studying target cardiac cells, cardiomyocytes, fibroblasts, endothelial cells, and different lineages of macrophages, recapitulate some of the beneficial readouts that we can estimate using these high throughput techniques into verifying the effects of the conditioned media and also exosomes. So that was the picture. So we injected cells, then using cell model systems, we recapitulate some of these observations with conditioned media and exosomes. From that, we became interested in trying to see what are the possible candidates. You know that a exosome can vary different molecules that potentially can interfere, so lipids, proteins, several types of RNAs, but we focus on microRNAs. So we assessed this in a rodent model, in a rat. So we interrogated about 700 microRNAs, and we detected expression of a little over 100 microRNAs in these mesenchymal cells. So using in silico pipeline, we end up selecting 5 candidate microRNAs that could be influenced or related to these phenotypes. So these 5 microRNAs, of course, were associated with different processes and so on. If we examine into myocardial infarction early, 2 days, 14 days, up to 1 month, they being modulated in this period. But there was one thing, 2 of them, these 2, they have never been associated with cardiovascular disease. And that's why we focus on them. So the 196, I will call 196, that was associated with these different processes, including mitochondrial functions, proliferation, and so on, and the 425, which I will not talk much about that. So going on, we using that same target cells approach and looking at different readouts to see if we could detect the influence of beneficial effects. We could reproduce also not only on the conditioned media, on the exosome, but now with the microRNA. So getting influence specifically on the 196, that's the one that we're going to dissect a little more, into very important phenotypes that was decreasing the amount of collagen, of fibrosis that is being deposited. So then the question became, what is the underlying mechanism whereby this microRNA exerts cardiac beneficial effects? So our first approach was to use these model systems and have as a readout global gene expression. So to do that, we used the 4 cells. We did a bunch of analysis, and the idea is to start to get a better insights on the processes, on the gene pathways, to then formulate hypotheses that can be directly tested for potential candidates. This is a busy slide, but I want to make very few points. So here you have the 4 cells. Myofibroblasts is the one that we're going to focus, and you see that the genes that are being downregulated, meaning the target of the microRNA, in this case, the largest number of genes in the myofibroblasts. Always you have the control cells, the control conditions on the left, and the treated with microRNA on the right. So everything here for endothelial cells, here for macrophages, M0s, and here for M1s. So then we start doing in the lab this integrated way of examining this data, in which we combine different data sets and allows us to see this in an integrated fashion, to try to get clusters of genes that are being modified. So you get here on the center, these clusters. Then here you have the different cells. They are coded in green, myofibroblasts, in red, endothelial cells, macrophages, M0s in magenta, and in green you have the M1s. And the idea is that some of the clusters can act in 2 different genes, some are specific to one, and so on. And you can, since we are going to focus on the myofibroblasts, you see that these regions are the most important. And again, you start seeing these networks that came up just from this enrichment that was done. So with fibrosis genes, fatty acids, and cholesterol metabolism, and so on. So you start to getting these ideas. Here's for endothelial cells, here for M0s, and M1 was very, almost no enrichment was observed. So then the next question is trying to do the target prioritization. So we're going to focus on the myofibroblasts. So we use different approach to test this idea, different data sets. So we select for the myofibroblasts the pairs of candidates that could be affected by the mirs, meaning that they have sequence that were amenable for the mir to bind. And you see that we selected 8 candidate genes, and for the other cells, different types. And one of them, this SLC 946, was common, the only one that was common among all the cell types. So here we see the expression of the mir on animals that have been submitted to infarct. As we expected, higher expression transient 2 days post-MI, 7 days. By 14 days, it's pretty much normalized. Then using in vitro assays on myofibroblasts. These myofibroblasts are extracted from rats that have been submitted to myocardial infarction. So you wait a couple months and then you get them in culture, and they have the properties of the myofibroblasts. And you can see that from the 8 genes, candidate genes, we pretty much reproduce the effect on 4 of them. This did not quite reach significance, but the most important, we got direct evidence of physical interaction. So doing pull down MIR 196 experiments. So not only you decrease they have the sequence that suggests interaction, they did decrease the expression, and also we got an evidence that there was physical interaction between them. So this is good because this allows you to now start proposing hypotheses to be tested directly. And the idea here is that this gene, SLC9A6, silencing would reduce cardiac fibrosis. So using these cell model systems, now you can first use the MIR and see what happens with the expression, if it is associated with reduction of fibrosis, but this could be just a simple association. So then you can do an siRNA against the SLC and see if it still reduces the fibrosis, or you can combine both and see if you have a double effect. So in the expected results would look like something like this. So again, you start building up still on the cell model systems to then go back to the more complex scenario. And here I show you what happens when you silence the SLC9A6 and fibrosis mediators, or meaning downstreams on this gene. And the first thing is that if we use the SI, you can reduce the expression, and then you take two proteins here in terms of expression of a matrix protein, important in the matrix production, also showing reduction, and here expression of the protein, again, another target for protein associated with fibrosis. And also we observed that there was a decrease in the proliferation of these cells. So once we get some of this data using these model systems, we try to go back to the complexity. And we are now, like others, trying to get more situations that are more similar to the in vivo situation. And we have been playing a lot with tissue culture that you probably have heard. So essentially, you get a piece of the ventricle, and then you work with a piece that's almost 1 by 1, 300 micron slices. If you can make this work or not. Maybe here. No. Well, it's a pity because you could see this working, but everybody's invited to come to the lab. So I don't think. Yeah, maybe here. So with this system, you can keep this up to 2 weeks, and you can also pace. So in terms of distension and electrically. So it's really pretty close. And you can either do perturbation in the animal. For instance, we can try to do a treatment with the siRNA or the microRNA in vivo or in the tissue. And we have another system that's not sophisticated as this one, but we don't have deformation, but we do have the electrical pacing. So in that we can keep up to 2 weeks in these systems, which is very, very nice to start doing complementary work before we go to the whole animal. So this is some of the preliminary results here. So you get an animal, have an MI, you take the slice, you cultivate that in that fashion. In this case, we transfect the slice, so not the animal, but the slice. So we could increase the expression of the MIR, and then we did an array analysis, and we got few genes modified, 55 down and 12 up. You can do some analysis, what's similar or not to the cells that we evaluated before. And the thing is that it appears that we're on the right track, but there is a pharmacological problem. We think that we are not getting much of inhibition yet. So we are now trying to improve this with different approaches, how we are transfecting either the animal or the tissue slice. But it suggests that altogether, this data show you that we provide evidence that the MIR-196 directly inhibits the profibrotic gene pathways, including this gene, and I didn't show, but the others also are playing a role, and these targets are now being explored to modulate the cardiac repair response post-MI in vivo, in using specifically the pig. Now, in the final minutes that I think I still have, right? Yeah, I will give you a taste of how we are addressing this endothelial dysfunction. Endothelial dysfunction is something very interesting because it's one of those things that everybody knows that exists, is important, is underlying of every disease, very hard to define. So we know that's bad, we know that indicates that the vessel is not working well, and until a few years ago, that was almost like a synonymous that if the endothelial cells or the vessel could produce a NO or not, or if they produce very little and NO when it should produce more. So this deficit of a NO production, it's considered the endothelial dysfunction. But it's a much broader issue because it has to do with all the roles that endothelial has in terms of maintaining the vessel, not clogging and everything. So, and we know that this is an underlying factor for a number of cardiovascular disease. We know also that a number of factors can influence the endothelium, like, for instance, the lipids. We can model this with OX-PAPC. The flow, if the flow is organized or disorganized, it has a major impact on flow. Inflammatory factors, hypoxia, and of course, most of the patients, cardiovascular patients, they have a cluster of this disease. They don't have only one factor. It's interesting because our research always, or most of the time, focuses one stimulus at a time, when in fact the patients often appears to you with several of these cardiovascular risk factors together. So then we start to try to model this in slides. You can control shear, you can control the pattern of flow. You can add, for instance, produce hypoxia with cobalt. You can mimic the inflammatory with IL-1 beta, and you can mimic the lipid with this. You can show that you activate the proper surrogate markers, and you have your system going. So using, again, expression as a readout, first thing that was interesting is 1 + 1 + 1 + 1 equal or different 4, meaning if I add each one of the cardiovascular risk factors, I will just get an added effect or I get something new. And long and behold, we got something that was not predicted. So of the 600 and so genes that were differentially expressed, 349 could be explained by the sum of the individual stimuli. But 271, almost 40%, falls in that category of emergent properties. So it's a property that does not appear when you have one single factor, but it's a new thing that occurs when you add things together. So this is the first thing that we demonstrated, is that these factors probably interact and create different components. So one major consequence of this is that if you're studying one stimulus at a time, you're not going to pick it up. Several things are going to be left on the side, because for them to appear, you have to have a combination of these factors. Then the second thing is that what's the hierarchy of this? Of course, this is depending on how do you set the stage to answer this question. But one way is that how many genes are differently expressed by each one of these stimuli, independent or when you combine them. And you can see that the hypoxia gave the least number of genes altered. Then you get the lipids, then inflammation, and most important, you got a lot of with the flow pattern. And of course, if you combine everything, you get not only some of the ones that you saw before, but the new ones. And so using this type of criteria, we did see a hierarchy in this response. So there is a lack of drugs that target endothelial cell dysfunction directly. And then the next question was, can a system multi-omic approach identify candidate targets for endothelial dysfunction? Similar to what was mentioned in the previous talk, we also are trying to do this multi-omic approach or trying to integrate different data. And in this experiment, we essentially combine the sequencing, the RNA-seq, with the epigenetic changes that are occurring on the DNA to see if we could combine both to have better certainty on the genes that were selected. And using this approach, we selected 81 genes, tried to do a lot of modeling, network analysis, to see what kind of network was that, and then we perturbate each one of them individually to try to get an insight in how that alteration on this network would have a consequence in 4 complex readouts of endothelial dysfunction. And that's my final 3 or 4 slides that I will show. So this is just what I mentioned. You combine then the RNA-seq with the tree approach for the epigenetics. You come with these 81 genes. You can do a lot of things trying to understand functions, properties, which transcription factors and targets, how they are sort of organized. But the most important thing, once we got those 81 genes, we selected 4 sort of complex phenotypes: cell proliferation, inflammation, ROS production, and cell permeability. Sort of complex readouts. And then ask the question, what is the influence of each one of those genes for this phenotype? And the most important thing is that we start to look at them combined. So we didn't look at them individually and had this aggregated rank analysis that allows us to identify 2 things that were not very obvious. So first of all, 26 genes, almost 1/3 of the genes, when we knock them down with siRNA, they somehow got the endothelial dysfunction worse. These genes in gray did not do much, and then the 31 genes in blue, when you were knocked down, they improved or they decreased endothelial dysfunction. So you see that even though we selected genes with the same logic, 81 genes that were differently expressed, have open chromatin, they were being affected, and so on, we sort of stumble in a process in which probably these genes are being controlled, but to sustain the cell on the pathological state. And if I knock them down, things are going to get even worse. But they're never going to be targets, therapeutic targets. While these genes, when we knock them down, somehow they decrease, they could adjust the network and improve the endothelial dysfunction. So that was the main insight of this paper. And as a whole, what I have shown to you, cardiac mesenchymal stem cell therapy is associated with improving tissue perfusion, eventually making more vessels, or eventually decreasing endothelial dysfunction. Cardiovascular risk factors affecting endothelial dysfunction in a hierarchical manner. So at least using this criteria of differential gene expression, and a system multi-omic approach uncover anti-endothelial dysfunction targets, which is the ones on the left, as well as a pro-endothelial dysfunction genes. The former are being considered candidate therapeutic genes targets for endothelial dysfunction, and we are now in the way of testing that. Finally, as a way of bringing things back to more complex, I mentioned to you the tissue culture, and before we go to animal, we are also modeling cardiac organoids either with single cell, only with cardiac cells, or most important, with the combined one, with the combined cells, fibroblasts, endothelial cells, pericytes, and eventually some inflammatory cells. We have plenty of data in data banks indicating from single cell RNA-seq what is the expected percentage of these cells in different diseases, and we are trying to modulate that, and we are trying to see if we can, number one, quantify cardiac fibrosis using this approach, which is pretty much straightforward. And most difficult, we're trying to envision ways of doing endothelial dysfunction assays also using this approach. Finally, I want to thank you for your attention and the people in the lab that have participated in these studies, our collaborators, and most important also the funding body. Thank you.
Thank you, Professor Krieger, with your nice speech. Someone has question?
Yeah, thank you for your talk. It was really, really nice. I have a specific question about your mRNA and the cluster of the genes that you described at the end. Did you detect some relationship with CTGF that has been mainly involved in scarring process and misregulation of scarring process to forward the fibrosis, and there was genetic factors that mainly control this scarring and fibrosis process?
We have seen only on the context of what is in the literature. The ones that we are focusing now and try to follow is that pathway that I mentioned that starts with that NH6 protein and also some of the other candidates. Those are the ones that we are following through, because the difficult part on this, as you probably know, is always the distinction between cause and consequence in this case. So going from this route, which is a pretty painful one, but at least on this we may get some insights. And if I may, what we learned from the endothelial dysfunction is that in these complex scenarios, probably a single target won't do much. And most likely, even though we have not shown that, but on the fibrosis is going to be the same. Because when we start from the microRNA and so on, I mean, it's that idea of fine-tuning. And so we have to eventually hit different things on the network to make an effect. So that's the biggest challenge now.
Hi, so you identified the microRNA 196A as a suppressor of fibroblast activity. And you also mentioned that fibrosis is important to fill the hole. So what happens when you suppress fibrosis during MI that you need to fill the hole with scar to the tissue? Or, I mean, you can actually protect or increase the damaging? Or are you thinking of like a combining treatment, for example, the mesenchymal cells with the microRNA that you have enough materials to actually have a functional tissue instead of scar?
Yeah. This is a fine balance. In the past, people have used drugs that, in fact, okay, you suppress the fibrosis, but you kill the patient, right? Because you don't have that. So when you go for the microRNA, as we were discussing here, you're doing fine-tuning. So that's why you should not expect major effects on a single target, because it's sort of having a global effect, like altering the setup, the set point. And so I don't think, even though we have not done the experiment, that any single one of these will be able to make the whole. But then the other question is, will be able to reduce to the extent that it will be beneficial? That's the major challenge. And as you pointed out, it's funny because we go around, but the cell seems to be the best thing, because it's amazing. I mean, it does, even though some of this route that I show here, I examine one of the or one class of candidates, microRNA, but the cells are producing, mesenchymal cells are producing tons of other things. So, but the thing that it's very difficult to make a product with the cell because variation, so on. So that's why we want to go to a small molecule. And but again, it's not only for therapeutics, because we are trying to understand better these pathways. But from the point of view that we started, I don't expect that a single one of these eventually will make the whole. The problem is that a single one may not make much either. So therapeutics. So that's why we may have to have a combined approach.
Really good, amazing talk. I just wanted to ask you, so you're making organoids from iPSC-derived differentiated cells, different cells?
Different cardiomyocytes, because as everyone has told here, the cells are immature. So I'm sticking with only the cardiomyocyte immature. The others are adult.
Those are?
Adult lineages. Yes. The only one that I use is the one that I cannot get from anywhere.
Okay.
So the cardiomyocyte is from the iPSC.
Right.
The endothelial, the fibroblasts, and so on are
Patient right.
yeah, adults. Yeah.
Okay. So have you tried to see an effect of any genetic polymorphism to, for instance, to differentiate cardiomyocytes with some gene variant that can change the properties in your system, in your organoid system?
We and others are dying to do that. The problem is efficiency and to be able to do that. I mean, this is a very expensive business, and we have to be very good at efficiency to try to start doing it. We have tons of patients. There's another line of research, as you are aware, that we are doing with cardiomyopathies that are a single disease, hypertrophic, dilated, arrhythmias, so on. So we have tons of opportunities, but it's very hard to establish for each one a cell. First of all, it's not a single. You have to have more than one. And it gets very complicated. So there are people that are trying to get very efficient, high throughput approach, low cost, but we are not quite there yet. But that would be ideal to try to untangle some of this. I mean, imagine the cardiomyopathy, hypertrophic cardiomyopathy. I mean, same family, same mutation, different phenotypes. Or I mean, you get one of these very well-known mutations that everybody assumes that is, you have you know that it's a causative one, and then you do a populational study. A lot of people are walking on the streets with that mutation and nothing else. So there's a lot of things that we don't know, and has to do probably not only with the environmental factors, but the genetic environment. And to test this hypothesis, we would have to have this type of approach, but we are not quite there yet.
Okay, last question.
Great talk. You show us that one of the targets of your microRNA, the 196, is a sodium proton exchanger that is important for endosomal maturation. So it's very important for the secretory pathway. I'm wondering, like, when you increase the expression of the microRNA, do you have, and you show us that it's a fact like cardiac fibrosis. Is it because you impair myofibroblast differentiation, or it's because it's disrupt the secretory pathway, for instance, by reducing collagen secretion? Do you guys have any clue about that? Thank you.
Not precise, but I have some clues. So it's not only because we do perform I mentioned here the myofibroblast that I removed from a rat has an MI, but another way to make an activated fibroblast is using TGF-beta or whatever. So and we have done the experiment in which you do the therapeutic or the preventive, and it does affect that, but not only that, because in a let's say on a myofibroblast, I also reduce the secretion of collagen. So it's probably affecting not a single.
More than one target. Cool.
Yeah.
Thank you.
Okay, thank you, Professor. So now I would like to invite all the speakers of this afternoon for the desk, so we can have a 10-minute talk about some new questions for everyone, and have an overview what we discussed today. Okay, so we can come all day of today afternoon. So it was you, Christoph, and the desk. So anyone wants to start?
So, Professor Krieger, I have a question about, do you think that the mesenchymal cells can be, its effects can be in part because it can be acting in place of the endothelial progenitor cell, or it has anything, it doesn't have anything to do with that?
Cannot exclude that possibility, but probably that's not the most important. Because remember, at the beginning of this game, at least in the cardiovascular, the idea is that these cells was like a joker. We implant that and would give rise to endothelial cells, to cardiomyocyte, this and that, and we would, like playing cards, right? We would solve everything. So even though they do differentiate into endothelial cells, I think that hardly will be able to explain the whole response.
Do you have data injecting the endothelial progenitor cell on in vivo to see its effects?
You mean effect?
Yeah, you differentiate outside.
Yeah, no, I have not done that. But I think in the literature you do have. So, but again, what people have not shown yet is that the cells that you inject will stay there for long. That's a major issue in this area. And I know that these cells that I inject, I will guess that will not be longer than 7 to 10 days. And this when I inject directly. And I know that I estimate. But I know for sure that 4 weeks later, I have no single cell there. So it's not there at all. So the other thing that people have not tried, it's multiple injections. So we are playing with different devices and things like that because you cannot open the chest or for that. And so that's one important aspect. But so far, we don't have strong evidence that the cells that you inject are there either because they have already been pre-differentiated or they will differentiate into something and they will stay there. It looks like they are coming to really alter the microenvironment and give a kickoff. Because remember, when you have a state of a war in the tissue, post-MI, for instance, the tissue is trying to recover, but it can't. Or it's doing something, but not enough. I believe that some of the things that we are doing is just pushing the system. So there's not much magic there. And the trouble is that the mesenchymal cells that we have tried, they are very good at that. And most likely because they are secreting, but they are not there. So they are not becoming something. I mean, they act for a while and then that's it. So the question is, can we improve that? So there are several ways trying to inject them on the pericardium so they would stay there and sort of a closed environment and producing goods. So we are trying different kind of things.
Thank you.
Let me ask you a question, Ricky. So when you identified microRNA 196, did you identify it as cargo from exosomes from your cells?
Yes. Because we isolate the exosomes. We use an array, and so we 700 microRNA at the time. We showed that maybe 120 or a little less than that were expressed, and then we use a lot of in silico analysis to come up with some candidates. Then we showed that the candidates, in fact, are expressed post-MI. And especially this one that I'm working, the 196, it's not expressed. So it's induced. So maybe it was expressed at some time and so on. But it's not expressed. And that's the challenge because I showed you that once I transfect the tissue, I can improve. But I'm going from 0 to somewhere. But I don't know if this somewhere is enough. Looks like it's not enough based on the experiment that I have done. So that's why we're trying to improve that.
I have a question, like a broad question, out of the box for all of you. With the rise of artificial intelligence, how do you think this AI era will impact in the development of research in cardiology and neurology?
So I think we had a taste about that on the multiomics arena. I think it's something that we're going to be able to integrate more complex data. From the time we've started doing proteomics, for example, about 20 years ago, 20 or more than that, 30 years ago actually, we were dealing with very few proteins, trying to connect, for example, some of the things. Also, for RNA, in RNA levels, and even in genomics, but the increase in complexity of the data generated, as more power we have, computational power we have, more we can progress in terms of integrating multiomics data, for instance, and then I think it's actually becoming more complex. Right, I think Krieger showed here some of the things that when you can integrate something, it gets very complex to understand. But I think this probably is going to be the next phase. Like now we can integrate, but how can we interpret all of that? Anyway, it goes that way.
Yeah, I think it's really affecting different arenas. If you think in terms of the hospital, like we are there, I mean, you can think of, number one, we are getting image analysis much easier. What was done before with modeling, now it's done routinely with AI. Then again, developing biomarkers, for instance, measurement of blood pressure. You need to put a device. This device, you stay 24 hours, have 15 minutes, you have to stop, something. Now we are placing a device. You did. So we are placing a device, and this is all done with AI. I mean, and the performance it's incredible. Then if you think in terms of a hospital like the Heart Institute, not all of you are familiar with, but it's probably the first hospital in Brazil that became paperless, meaning you have electronic health records, and since 2002. So we have about 1.5 million people in our data bank. And some of these patients that I mentioned, like heart failure patients, they can keep coming to the hospital for a decade or so, and so on. Some have died, and so you have then one day this guy shows up in emergency room. He has been a patient of the hospital for 15 years, and you are there, the attending. You get the patient. Where do you start? You have all this data about. So what we are doing is that there's in these electronic health records, you have a number of things that are already structured. But when the patient comes for a visit in the outpatient clinic or an emergency room, or when he's inpatient, there's a lot of things that every day someone goes there and writes something, even if you write digitally, but it's written. So that's not structured. Now we use LLM to get all that. So, and we take a lot of information that was completely lost from things that were happening, from diagnoses that were being made, from drugs that were taken, and now we are making all these available. Now we are preparing, for instance, a summary statement for a patient when he shows up in the emergency room, one of our patients that even if he has 15 years of history, it comes up right there to you. I mean, this is done like that. Yes. Yeah, it's not yet in place, but we are testing that. It's amazing. I mean, what you can do. Then, for instance, analyzing X-rays signals. We have algorithms developed by ourselves look at images, because there are a lot of companies already sell the device and the way to analyze. No, ours is based on a picture, and even the picture, because there are some contests. We just won one in Germany, the Physiogen, that you have to have coffee on top, or eventually the paper is not clean, and so on, and to see if it works, and it does work. So sometimes it's not to replace the physician, but it's a classifier. If you go to the 10th floor, you see there is sometimes there either an X-ray, chest X-ray, or an ECG. And ECG is every machine in the Heart Institute is connected to that. before anyone has seen the ECG, is already there, declassified. So even if it's not completely 100% correct, it has classes of, for instance, prioritize. So you get to see this patient first and so forth. So this is having a tremendous impact. And the final example, I think everyone here that probably have hundreds of examples. For instance, the great difficulty of understanding the relationship of the sequence of a protein and function. I mean, until a few years ago, we were only dependent on crystals. Now, I mean, it's amazing. I mean, what's going on? So it's a different way of looking at it. I mean, it's going to add, empower, I think, the researchers very much. Yeah. Yeah. Well, like everything, it's a swing, right? For some people, it's going to be, oh, this is the best thing. No, it's not good at all. I mean, the swing is going to stop someplace, but I mean, it's definitely here to stay. Yeah.
Someone want to make a last question? Oh yeah, of course.
So yeah, a nice discussion about the AI. I completely agree what you are suggesting, that, you know, like data analysis, image analysis, many things are streamlined, biomarkers. But let's say if I ask a question, you know, on the other spectrum, that what are the pathways of schizophrenia? You know, I asked ChatGPT and they will give some hypothesis. So I think we need to test those in the experimental system before we can take it forward, right? So on one hand, yes, it's making human jobs simpler. On the other hand, it's giving some suggestions which we then need to go to the lab and verify. So.
Okay, so I thank you everyone here, all the speakers and all you, and we see tomorrow at 8:30 in the same place. Thank you.